Hi, Madhu. Nice poster.
Hi,
Or nice background.
Hi, Dennis. Thanks for joining us.
Hi, Dennis. Okay. Oh, you guys have the same background. I'm jealous now.
Yeah, it's the corporate view. Yeah.
Yeah, very good. Okay. Very good. I just have my usual room where I sit all day long. Well, I'm just going to close the door. And I'm not quite sure. Might wait an extra minute or two. Since it's hard to see, we have a lot of people, which is great. Now I can't actually find everybody. Oh, there's Rianne. Okay, perfect.
Hi, everybody.
Thanks for joining us, Rian.
Oh,
Okay.
It's an absolute pleasure.
Yes, great. Okay, so I think we will get started. It's 1 minute after. So I wanted to welcome everyone who has joined or is joining and connecting to our 9th annual research day of the McGill International TB Center. I'm very pleased to welcome our speakers, TB Center members, trainees, and all other TB aficionados, with or without the official WHO World TB Day logo in their background. We have a packed agenda, opening remarks by the director of our research institute, who I'll introduce shortly, followed by 4 speakers. Then we have 3 research presentations by trainees, followed by awards and closing remarks. I just make a quick note to those who looked at the original agenda that was sent out some time ago. Dr. Soumya Swaminathan, Chief Scientific Officer at WHO, had been scheduled to speak, but a family emergency arose and she could not attend. And Dennis Falzon, also from the WHO, has graciously accepted to speak in her place. So without further ado, let me introduce our lead-off batter, so to speak, Professor Rianne Toos, who is the Executive Director and Chief Scientific Officer of the Research Institute of the MUHC. Born in South Africa, where she did her medical degree and PhD. She then did a postdoc fellowship at the IRCM here in Montreal, just up the street from the chest, as it turned out, for about 9 years. I hadn't realized, actually. Then she was recruited to Ottawa, where she was a Tier 1 Canada Research Chair, held that in hypertension. And then from there, she was stolen to by Scotland, and it was at the University of Glasgow. And finally, she was recruited back to Montreal. So we're very pleased to have her back here in Montreal as as I said, the Executive Director and Chief Scientific Officer of the Research Institute. So Dr. Toos, please take it away.
Well, Dick, firstly, let me say how just delighted, honored, and privileged I feel to be here opening this wonderful meeting that you've put together. I was reading through your mission statements. I went through the many presentations that you shared with me in terms of what you do in the center, and it's truly inspirational. And I congratulate you and your team on the innovation and pulling together so many bright minds and doing such fantastic work in the field of TB. Indeed, I did my medical training in South Africa, and let me tell you, we had TB hospitals in those days. I hate to say, I saw too many patients with, untreated TB and terrible devastation. So it's just wonderful to learn of the amazing things that you are all doing, and especially through this incredible initiative through the World Health Organization. So wishing you all continued success, a wonderful research day. And I'm just so happy now to be able to introduce our guest. And indeed, we feel truly honored to welcome Dr. Dennis Falzon, who works at the Global TB Program of the World Health Organization in Geneva, Switzerland. And I understand that before joining the World Health Organization in 2008, he was a very successful medical epidemiologist at the Euro TB Surveillance Project in France. And earlier before that worked in public health in both the state and non-governmental sectors. Dr. Falzon is currently leading a team working on TB prevention, research and innovation, and he's certainly been involved in the guidance on the treatment and management of TB disease and TB infection, and very importantly, TB prevention, integrating digital technologies in support of the Ntp Strategy. Of course, novel methods, innovation to disseminate guidance and research on TB innovations is absolutely critical, spreading the word. So with that, let me welcome Dr. Falzon. And again, let me say, I wish you a fabulous research day, and thank you so much.
Thank you very much, Rian, for these kind introductions, and thanks, Madhu, Dick, and your colleagues for inviting WHO to open this important day. As you know, this is the World TB Day, which we commemorate as the day when Robert Koch announced to the community the discovery of Mycobacterium tuberculosis. It's about 140 years to the day today. Okay, so let me see if I can manage to open my screen, the right one, that is. Okay, so can you see that?
Yes.
Can you see it in full now?
Yes.
Yes.
Okay,
Yes.
I'm getting better at this. Okay, thank you very much. So my presentation, and I will try to stick to 20 minutes, and stop me if I don't, will be in 3 parts. So I will discuss a little bit about the impact of COVID on TB as we have described it to date, and then a little bit about what we envisage for the future, and something about the response, and concluding on that note. So just by way of start, I'd like to make the point that TB remains a hugely important global public health problem, both in developing and the developed world, as we see very often changes in the global geopolitics can easily flip TB as on the agenda. So a quarter of the world's population is estimated to be infected with TB bacilli, which presents a huge pool from whom TB, active TB, or TB disease as we call it, can emerge. And we estimate that we have about 10 million new cases every year. And in the last estimate, during the first pandemic year of 2020, we had about 1.5 million TB deaths estimated. So just to put the in perspective, TB remains the most important single bacterial cause of death in the world. So it used to be the single infectious cause, and but it has been trumped by COVID. And very, very probably once the pandemic recedes, it will regain this position of #1 killer. Okay, the second point I wanted to make is about COVID-19. So COVID-19 has now been with us for 2 years at least, and it is not yet over. So it keeps bringing surprises. You see, it is all mostly with this trace, as you can see, on the going down. These are the number of deaths. But if you look at the latest number confirmed deaths, we are at sort of the number of deaths that we had during this first year of the COVID, for much of the first year of the pandemic. So still an important cause of death, an important cause of hospitalization and of sickness, of course. There are a number of potential points of interaction between COVID-19 and TB. First and foremost is something that has become very evident to everyone over the last 2 years: disruption to efforts to control TB, be it in delaying or missing diagnoses, as well as the impact it has had on monitoring and evaluation, the failure to start or to continue TB curative treatment or else TB preventive treatment, the effect on BC vaccination, and other programmatic components of national TB programs. Then there's also the non-negligible effect on the drivers, be it disruption, for instance, to provision of antiretrovirals for people living with HIV, poverty, the impact on nutrition, on smoking, on alcohol, use of drugs, migration, and this issue on social protection, on universal healthcare coverage. So all of these have been stretched, have been challenged by the COVID pandemic, and they will have an indirect effect on TB. There has been also, presumably and very plausibly, an effect that the change in mobility, change in mixing patterns of the population, use and non-use of masks, might also have affected the community transmission of TB. And finally, the synergy between COVID-19 and TB in the same individual. We'll speak a bit about all of this in the coming slides. So we have seen these traces for a long time. I'm not going to labor you with too many of these type of traces by country, but this is the trace in India, and black line is the TB notification patterns from January 2020, as you see, just before the pandemic hit. And you can see in black the trace in the public sector, and in red in the private sector. Both of them took a plunge when COVID related lockdowns took place in the first year, just after the pandemic started, and then also into the second year, as you can see. So these are important indicators of the impact that COVID has had on TB programs. This is the result from a survey that WHO repeats. It's in its third cycle, and this refers to the last quarter of last year. And this was asking countries that from the communicable disease programs, how many of your programs have been reporting disruptions. And as you can see, TB diagnosis and treatment is the first of the pack over there. So about 49% of countries have reported some level of disruption. Just to point that the effect is still ongoing. We're still in the midst of it. Even early on in the pandemic, the early predictions that there will be levels of disruption. This is some modeling that my colleagues in the global TB program had done at the very beginning, about 2 years ago, and it showed that even modest, sort of seemingly modest reductions in the case detection of about 25% drop for about 3 months would increase TB deaths by about 200,000. And we've seen that in 2020, we had about 100,000 deaths more than the previous years, okay, which is an important development, given that TB deaths had been on the decline for since 2005. So it was the first time that there was this uptick in the amount of deaths from TB. And then this dramatic drop that we reported in our global TB report last year, 18% drop between 2020 and 2019 in terms of TB notifications, a drop that was registered in all regions of the world and in most countries with the highest TB burden, as you can see in these bars representing each of these 30 countries with respect to the vertical black line, which is the position in 2019. I mentioned this already about the increase in TB deaths. This was when you consider that, for instance, deaths from HIV continued to decline in 2020, TB deaths went in the reverse direction. So an important development. So notification is one thing, of course. There has been impact on other components of TB programs. This is TB preventive treatment activities in the Philippines, for instance. This important drop of about a half compared to 2019 in the reporting districts of the country. And again, an effect on BCG vaccination. This is from India again, but it was also registered globally, and we reported on this in our global TB report. As I mentioned, COVID-19 has had an impact already on a number of these determinants of TB, be it other comorbidities, including HIV, malnutrition, and also the effect on the health services. When it comes to the effect of COVID and TB directly in the same individuals, this is still a question that we are looking into, and with the help of McGill colleagues as well, we are doing an evidence review to look at evidence for the synergy between the two conditions in the same individuals. But you can see again from a study that was conducted in primary healthcare centers in Western Cape, South Africa, you can see this effect in COVID patients who had TB in the past or concurrent TB, where there was a statistically significant increased risk of deaths in both groups, more pronounced in those with concurrent TB. Okay, let's look a little bit on the predictions. Of course, predictions always come with some level of uncertainty. We released these in the last report in October last year. These traces show in blue the expected incidence of TB into the coming years, compared to a baseline value from before the disruptions had started, assuming no disruptions. And you can see that in all of these countries with high burden, these are selected countries, but in all of them, there is this increase in incidence that is anticipated. Similarly, in red lines, you will see mortality rates, TB mortality rates. And again, all of these countries with very different epidemiological patterns for TB are expecting a substantial increase in deaths. Okay, these are data from the from the World Bank, and these are relating to the output, the financial situation in different groups of countries. And you can see that in both the developing and in the rich world, there have been this huge adversity in 2020, and the recovery has been much slower than was expected. I think, sorry, I'm moving very fast here. I think it's really, this is really important because the effect of this is expected to be quite profound on health system resilience and also on the capacity of patients themselves on poverty, and that is a known fuel for TB incidence. But also in the rich countries, I think this just demonstrates that countries that are usually donor countries might also be compromised further to deal with their own sort of domestic problem before they can, and with so the disposable income part in these countries might be reduced. Okay, let's see about the last part of my presentation, something about response and about what we've been doing. So this is the trace of the post-2015 Ntp strategy. This is the anticipated reductions in TB incidence that we wanted to have from 2015, where the overall incidence was at about 100 per 100,000 globally. And we want along with all the TB community, we aspire to get down to levels that we see in rich countries today by 2035 at the latest. And of course, this trace, I superimpose this reddish line or reddish bar here to represent the start of the pandemic. We have experienced, of course, adversity in these in decrease. This year we will be estimating a bit better how much the damage has been continued into 2021 in order to see. But we really want to be back on track with new tools, and I mean, I think there are signs of hope as well that with the pandemic and other challenges that have happened might help us to get faster towards this aim. The Ntp strategy, for those who may be not very familiar with it, is an all-comprehensive type of strategy which looks at not just targeting TB, but also looking at sort of the upstream determinants of tuberculosis, poverty, universal healthcare, etc., and trying to address those, and also to trigger research in this third pillar. Something about the COVID response from WHO. I'm talking now from the people in emergencies. This was immediately one of the strategies that was launched and recommended to have a multiple sort of approach to the pandemic. And as you can see in number 9 here, strengthening of the essential health services was a key component in all of this, because we are all keenly aware that when pandemics such as these and cataclysms occur such as these, is very often the deaths that are driven by neglect of essential programs, and TB is one of these cardinal programs. These are 2 key documents that I put here maybe for people to look into, but to consider about, especially this document on the right here about maintaining essential health services, which describes as well on TB specifically about the different phases and monitoring for different indicators. also late into 2020, we had the UN Secretary General making these recommendations on trying to recover from the pandemic situation. I think there were some openings as well, some opportunities coming up, because the extremity to which COVID put us also triggered some things that had been recommended for long but not implemented, such as the increased focus on home-based care, on household contact investigation, where we wanted to have more action for TB, and this is a very poorly developed component of many programs, and it needs to be done. The importance of electronic health records and digital adherence technologies, and also e-learning for capacity building, these took on a new more importance in the within the programs, and also the use of shorter treatment regimens, be it for the treatment of multi-drug resistant TB and also for TB preventive treatment, because these are regimens that have been recommended relatively recently, but which have not been well implemented in many countries. So there has been increased insistence on trying to get key drugs like bedaquiline, like rifapentine, for treatment in the different places. I think one of the things that became obvious during the pandemic is that if we want, we can gather data globally at fairly timely rhythm from countries. You know, these maps which are updated daily. So I updated this morning. Some data came in yesterday for this map, just to impress on the importance. This is just showing the coverage of vaccination. So it shows two things. But the point I wanted to make really is about the spurt that this the COVID pandemic has given to monitoring. So this has also helped us to spare countries as well to send data on quarterly or monthly on monthly rhythm. So we are getting data from many of the high burden countries, and you can access these via the links that I'm showing here. So you can see them for many countries, and they will show you until the last quarter or last month the data. And you can see, for instance, these are data from Ethiopia. In blue, you can see the trace for 2020 going beyond the 2019 baseline. And again, in green is the 2021, again, completely below the 2019 baseline. So still a bit of bad news. And unfortunately, this is the situation in many of the countries still. This is the number of countries that now are showing in green here are the countries that are known to have all of their TB notification data available nationwide in electronic case-based format. So this map over the last 10-15 years has gradually increased, and thankfully we are able to get a much better handle on what is going on in many of these countries. And in other shades are partial data for, let's say, MDR-TB patients or part of the country, etc. But a huge improvement, I think, and this is to some degree had been helped by the pandemic itself. Another point I think of importance has been the importance of testing, right, for COVID-19, and alongside testing for COVID-19, because many of the people who present for COVID care present to the frontline health services who are also dealing with other pulmonary diseases, including TB. You may be aware that in many places the same staff who are dealing with COVID-19, with TB had to suddenly displace all of that and start dealing with COVID-19. So last year we worked with colleagues in the Global Fund to come up with guidance on doing the dual testing for TB and SARS-CoV, defining 3 use cases when people might present and present opportunities as well to be screened for TB when they come for COVID testing. And on the right is another document that is complementary and came out at the same time as the briefing note from the Global Fund, and provide some implementation tips and very practical points for the implementer. Again, some other important things I think has been the refreshed funding for what is called the COVID-19 response mechanism. and this was a funding that's channeled through the Global Fund. You can read more about it, and there's a lot of documentation from the Global Fund accessible via that link. And also on the right, a collection of case studies from many countries displaying innovations that they've done to try and keep TB on the on the forefront during the pandemic, and we will be issuing very soon a new update on this, which will again speak about more recent developments like dual testing or this bidirectional testing and the use of digital technologies. So one other thing that that we released as well last year was this knowledge sharing platform. This had been a long-standing sort of request for us to concentrate all of our guidelines, the handbooks, and now our training modules, the new training modules that came out from late last year in one place, and this has happened now. So these are available, and this has also been again pushed by the requirements of COVID-19. It's becoming increasingly difficult to send paper documents, right, or less favorable nowadays to send paper documents that expire very fast, and more people are becoming adept at using electronic tools. So it's available in mobile format, and we keep updating it. We will shortly. There's already a French interface. We'll have all different language interfaces again. And we are also going to release an offline tool so that you can install the same knowledge sharing platform on your computer, on your laptop, and have it available, or have most of the functions available, even when you are not connected to internet, which is the situation in many places where these guidelines need to be used. We've also updated regularly this information note. You can see it here. We've been updating this for last since this start when we first released it in the beginning of 2020. And we're also now doing, as I mentioned previously, some evidence reviews for potential synergies between the two conditions. So and with the help of our colleagues at McGill, we're close to finishing this evidence review. We get common questions, and we try to answer some of them, that information note that I mentioned, but I think it's always worth coming back to the evidence was being published, and there's a lot being published, as you know, on COVID-19, and to see, for instance, to try and answer questions with data on should TB patients be prioritized for COVID-19 vaccination, right? I mean, what is the evidence base for that, and when and how to do this bidirectional testing for the two conditions, or what are the best therapeutic or preventive options for people with both conditions to reduce severity, improve outcomes. And again, a big question about, does COVID-19 predispose to TB? Should TB patients be offered screening? Should they be offered TB preventive treatment? Will the pool of COVID-infected patients and people in the world, right, which is measured in percents, will they be more vulnerable to TB in future? So these are, I think, key questions. I don't think we'll have the answers for all of them, but I think by putting the questions, we will ask the researchers in a way to start preparing data in that manner. So by way of conclusion, I think COVID-19 has reversed many years of progress in provision of TB services and reducing the TB disease burden. The impact will outlast the pandemic itself. The global TB targets are unfortunately mostly off track, although there are some country and regional success stories on which we can try to build ways of extrapolating these successes elsewhere. Addressing COVID-19 adequately will challenge competing needs to maintain, continue to develop essential services like TB, and the catch-up post-pandemic will be influenced by the global socioeconomic recovery. We need mobilization of funding to prevent that vulnerable populations get increasingly marginalized, and finally, stronger evidence for biological programmatic interactions needed into the future. And with that, once more, I thank you for inviting WHO to this important day. I thank also my many colleagues who put together all of these slides, and many other people, the patients themselves, and I welcome if there are any questions for discussion. Thanks very much.
Great. Thank you, Dennis. Wonderful. That's indeed a rapid fire review of a very major phenomenon. So I wonder, I've asked people for questions through the chat, or you can simply raise your hand. I guess one question I had, Dennis, is what about if the pandemic doesn't end? It just kind of goes on and on and on. We're now into year 3 really, and there seems to be no clear end in sight. So what, I'm sure you guys have discussed this as well, and what will be your strategy then?
Yes, I mean, that's an important point to consider. It could be, of course, it could become like an endemic condition. I mean, well, the future will prove me right or wrong, right? It will become an endemic condition, and that hopefully it will stay a vaccine preventable condition, and we can start to vaccinate against it, a little bit like whooping cough, right? And deal with it like that. It could be another condition that gets transmitted in waves, like influenza. So from one year to the next, we need to revise our vaccine strategy and do mass vaccinations, in not just at birth or like an EPI type of strategy, but doing mass vaccination for people who are most vulnerable, like we do with flu today. So there are all of these possibilities, or else, I mean, it could be replaced by another type of coronavirus or another type of virus.
Of course. Okay, great. So I know Madhu's kind of been our specialist here at McGill on COVID and TB, has written and talked a lot about it. So Madhu, I don't know if you want to make a comment or have a specific question.
No, just to address your point, Dick, about this being far from over and likely to continue and particularly devastate the global south, because the global north is fairly vaccinated and we should be okay, barring anything disastrous happening with another bad variant. But 3 billion people in low and middle income countries haven't had even their first dose. And that's why I feel that all of us working in TB must advocate for vaccine equity, because vaccinating the world is our path out of this mess. It's not just for COVID, it's also directly benefiting us in TB. So anything we can do to hold our leaders accountable for sharing doses, for backing the TRIPS waiver, and for sharing other resources like the antiviral medicine will only be good for TB in the long run. Thank you.
Right, right. Same for TB as for COVID, absolutely. And Marcel, I don't know if you wanted to.
Yeah, well, Dick asked, what if COVID doesn't end? And another related question is, we've seen the declaration, the COVID pandemic. What's the definition of the end?
Yeah, that is always a difficult thing to define, right? Because it could mean different things in different settings, and signaling the end of a pandemic might, of course, disadvantage places which have been late in the pandemic, maybe, and where there's still a lot of reserve for the thing to become more of an outbreak and have more cases there and more disease and deaths. So so it is I think we'll need to see when the public health action needs to change into a different format, right? As the disease evolves, we need to keep monitoring the emergence of new variants of concern, I think, because these will be, as we've seen, some of them have taken pandemic sort of proportions in a very short period of time, and that's an important consideration. So it is not a simple sort of answer with that you can put on with a threshold that is reached over a period of time. I think the end of the pandemic has to be declared in a more nuanced fashion and in a way that is beneficial to people when one announces it.
I guess, so Dennis, you said that there are some success stories. So maybe you can just tell us, is there a program that has managed to somehow either build on COVID or work with the COVID control and integrate it into TB control in a way that they have managed to really preserve the key activities of the program. And I wonder if you have an example or two that would be a more positive message there.
Yes, so I think and some of this will be in the next version of this report on the innovations. I think the area of dual testing, I think, or what's called bidirectional testing, is an interesting one, because I think that is where we have uncovered, let's say, in some of these inner cities areas in Southeast Asia, for instance, where there are populations which are independently of the pandemic, but which are sort of at risk of TB. They're very high burden TB, and there's a lot of sort of unmet need of TB in those or untreated patients in those situations. And I think the implementation of these efforts to do dual testing has been very beneficial. Very often in these populations, we found prevalences of TB of about 1%, or even more, which would be sort of the way above the usual threshold that we use to recommend screening in a given subpopulation. So I think this has been an important one. I think we cite some examples here in the Philippines, for instance, in parts of Manila, where this has been done. I think this has been an important thing. I think the other important thing, and this map sort of points it out, is about the vaccination, right? The vaccination for COVID, because I think this, again, it's an important development. We've seen countries that could advance very fast. We see, for instance, India here, right, amongst the countries which has an increasing amount of total doses administered per 100 population, which is pretty high. So if countries like these can move the vaccination, right, and push vaccination for COVID, they also, in a way, sort of show that they can activate and to try and bring up or remove the disruptions that they had been experiences for a very long time in the TB area, so that bringing the pandemic to normality would cut down on the sort of disruptions to mobility, and again, sort of the TB patients and that trace, as you can see from last January now, has sort of trying to normalize. And I mean, of course, India, there are other countries, but contrasting to the other countries that show in red over here, but there have been successes in those other areas. I think as well the development of the COVID vaccine itself has reopened, the hope for other vaccines for TB in particular, for maybe for HIV as well. So again, I think these are some of the sort of non-negligible successes that may have been pushed forward during the pandemic.
And again, I don't want to go on too long, but it does seem as though public health has enjoyed kind of a higher profile and presumably more funding for public health in general. Maybe they stole it from TB, but I think a lot of places there was simply more funding, more jobs, more people, and the whole area was viewed as, more important by many governments. And I wonder if you're tracking that as well.
Yeah, I mean, to varying degrees, right? I think some countries have, of course, taken it more seriously than others, but there has been, certainly in the media, I mean, public health has become more important. Sometimes negative publicity has pushed it as well, but still, it's. I do agree with you that there has been greater importance because of the crisis that it's caused, because of the impact it has on the economy. So it has driven countries to try and act, I think the tricky part will be now during the recovery, when we go back, right, to try and reinstitute normality, to try and these patients who have been waiting for non-urgent interventions, for instance, surgery or whatever, to cut down on those waiting lists. how to make sure that the importance of of diseases like TB, which is always as kind of Cinderella, and other public health neglected TB disease, the neglected tropical diseases, how we can protect them, right, and make sure that they get their, they keep on the agenda. I mean, WHO has, as you know, has this promotes this all the time, and in many of our communications we say this, but it is something that we need to keep on the agenda about the importance of healthcare system resilience, the importance of universal healthcare, social protection, etc.
Great. Okay, so Dennis, thank you. I think I don't see any more questions coming in. So I think we'll pass to the next speaker. So we're moving from the global and very macro view to the granuloma. So a very, let's say, micro view. In some ways, this reflects the McGill TB Center, I could say, just to promote ourselves for a moment, Joanne, before I let you start speaking. In that, we have a center comprised of investigators that indeed span the full spectrum from macro and global, people like Madhu to people like Marcel and Maz, who are very involved in some of the basic fundamental mechanisms of TB disease. So I think Marcel is actually going to step in to introduce you, Joanne. Maz is on sabbatical, it turns out, in Nijmegen. So fortunately, Marcel is there.
Thank you.
All right. Yeah, it's a great pleasure to introduce Joanne, and she's already told us that she wants to come back. And the first comment she sent in the chat is, where's the good food? Briefly, Joanne is originally a Californian. She did her BSc at UC Davis and a PhD at Berkeley. She did a postdoc at the Scripps, and then she went east, and she did training and worked as research associate with Barry Bloom at the Albert Einstein College. Many of you know Barry from his advocacy work in TB, when he went from a wet lab scientist to a dean of public health. And at that time, Joanne wrote some seminal papers about the things that we all know as facts, but they had to come from somewhere. And if you look at the literature on how do we know interferon gamma or TNF matters for TB, you'll find Joanne Flynn articles about that. She's had major contributions to understanding of TB and the granuloma. She's migrated across animal models and currently is leading a very productive non-human primate research group that allows us to look in some ways directly at comparisons between humans and primates and have a better understanding of the biology of infection and disease. All kinds of awards, former president of the American Society of Immunology, among other things. So Joanne, thank you for joining us, and you will be invited back in real life. We look forward to your talk.
Thank you. I love Montreal. Thanks so much for that introduction. I will attempt to share my screen, and let's see, how did I do? Is it in, not yet, it's trying to be in full screen mode. Is that, can you guys see full screen mode?
Yeah, can
Okay,
see
great.
full screen. Congratulations.
So, so thank you again for this invitation on this, most inauspicious or auspicious day in the world of TB. This will be a complete 180 pivot from our last talk. As Dick said, we will be delving into the granulomas. So I think a lot about host pathogen interactions and how people or monkeys control or don't control TB. And it's first important to remember that all the critical host pathogen interactions in an established infection occur in this complex microenvironment of a granuloma. And here's a monkey granuloma that looks a lot like a human granuloma, and then just sort of a caricature of that. So as Marcel said, we work in non-human primates because we feel that they are the model that best recapitulates humans, but that you can actually get a lot of samples from, including individual granulomas in the lung. So I just wanted to briefly mention some of the tools and technologies we developed over the last 20 plus years to work in non-human primate TB. So we use barcoded MTB. So every individual bacterium has its own DNA barcode, and I'll come to that at the end of my talk to just show you how we use that information. We use various macaques, cynomolgus macaques, rhesus macaques. I think everything I'm talking about today is actually in cynomolgus macaques. We do serial PET-CT imaging, which allows us to, each blue arrow here points to a granuloma, and then we can watch how those granulomas change over time, which there can be new granulomas, there can be resolution of granulomas, and there can be granulomas that go very bad and turn into consolidations or cavities. We can obviously get serial blood and bronchoalveolar lavage. And what we've been able to do is to take that last PET-CT scan we do and then map it onto the lungs at the necropsy, so that every granuloma we take out, we have a whole history of when it appeared, what it might be doing over time, and then we can analyze the granuloma in a variety of different ways, including quantitative microbiology, immunohistochemistry, multidimensional flow, and single cell RNA sequencing, et cetera. So this gives us a lot of power to understand what happens over the course of infection and what individual granulomas might be doing. So just as a sort of preface to the rest of what I'm going to talk about, in this graph here, each column is a monkey. Each individual symbol is a granuloma from that monkey, and the y-axis is the number of bacteria, live bacteria per granuloma in a log scale. On the x-axis is the time post-infection that monkey was necropsied, and in weeks. And so what you can see is that there's a huge range in the bacterial burden in granulomas over time within an individual animal. But by about 10 weeks post-infection, although there are still granulomas that might have a lot of bugs, we start to sterilize some granulomas. And overall, as you can see in this graph down here, where we've calculated the mean bacterial burden per granuloma per monkey, so each dot here is a monkey, you can see it isn't until about 10 or 11 weeks where we really start to see a substantial decrease in the bacterial burden in the granulomas. And then this continue on into much later, in 6 months post-infection. So very early on, it's actually the time when the monkeys probably have the most bacteria in their granulomas, but yet they're not sick. So they are trying to contain it, but it takes a very long time to start to kill the bacteria. So what that means, we think, is that whatever is happening immunologically means that the bug has a chance to get a foothold in the lung and grow into, up to 10 to the 4th or 10 to the 5th bacteria per granuloma. And then it just takes time, and we assume this is the adaptive immune response taking a long time to be generated, and I'm going to show you some data on that finally you're able to kill the bacteria to some degree. So how do we actually take this idea of various outcomes of granulomas, and how do we actually start to interrogate what makes something successful, a successful granuloma or a permissive granuloma? So successful would be killing the bacteria and resolving, and permissive would be the bacteria continue to persist or grow and then disseminate into form new granulomas, which can lead to active TB. So we've taken a number of approaches to this, and I'm just going to focus here on one, which is the single cell RNA sequencing we've been doing in a fantastic collaboration with Alex Shalik and Sarah Fortune. And here are the people, Travis and Hannah and Constantine, who have really contributed substantially to this work, but it's the work of a large number of people in all of our labs. So in our initial experiment, which was just 4 macaques, we infected them with a low dose of MTB, so 15 CFU. We do serial PET-CT to see when the granulomas show up. We then use that scan and we isolate individual granulomas, plate them for bacterial burden and chromosomal sequencing, which I'll mention in a second, and dissociate and chose 26 random granulomas for single cell RNA sequencing. It's all done in the BSL-3. So here are our 4 monkeys, and here are our randomly chosen granulomas, because we don't know how many bugs they have until 3 weeks later. So we just randomly chose them. You can see there's a range of bacterial burdens and also some differences between the monkeys, which is very common. Monkeys are as heterogeneous as people because they're completely outbred. So then what we did is we took these granulomas and we binned them based on bacterial burden, so the number of live bacteria. You can see we have two groups, a high bacterial burden group and a low bacterial burden group. Then we do what we call CEQ, or chromosomal equivalence, and that tells us how many bacteria were ever in those granulomas, because the DNA from MTB takes a long time to go away. So you can assume that CEQ means live plus dead. And what you can see is that the high and low granulomas have similar, but not quite equal CEQ. So what we interpret that as is this is how high the number of bacteria grow in a granuloma. You can see it's a little bit lower in the low granulomas, but not significantly. But then when you compare the number of live to the number of total bacteria, you get a measure of killing. And so in the high burden granulomas, we're getting less killing than we are in the low burden granulomas. And then what we did is we took a look at the PET-CT data, and it turned out that the high burden granulomas, shown here in red, are what we call early or high burden. They all showed up in the initial infection. So we can reliably see granulomas up 4 weeks. Those are those that initially established. And the lower burden granulomas are those that didn't show up until 10 weeks post-infection, as the result likely of a dissemination event, although it could be slower growth of that granuloma. So when I talk about high burden and low burden, I'm also talking about early, which is high burden, and late granulomas. So this is just our U map of all the different cell types that we see in granulomas. It's actually a lot of cell types, some you might not expect. And these are just our designations. And then we determine, based on the bacterial burden, which types of cells positively or negatively correlated with bacterial burden. So if we start with those that are positively correlated, in other words, they're higher in the granulomas that have higher bacterial burden, it's kind of interesting. So it turns out to be plasma cells, which I would not have guessed, endothelial cells, a little bit of fibroblasts, and mast cells. And I don't think any of us really could have guessed that there was such a large proportion of mast cells in granulomas. But you can see they're primarily in the high burden lesions. And then again, I think no surprise, what correlates with lower bacterial burden is NK cells, T and NK cells. So they group together here on this map, and it's this big blue thing that looks like Africa if you're looking at the world. And so you can see that there's a negative correlation with bacterial burden, which means you have more T and NK cells in the lower bacterial burden lesions. So we wanted to, so we've done a lot of things, and I'm going to sort of cut to the chase on one story only. So we looked at the T and NK cells, and so we subclustered them. And we ended up with a number of different subclusters, 6 cytotoxic subclusters, a big subcluster called T1, T17 that I'll talk about in a second, and stem-like cells, proliferating cells, Tregs that you'd expect. And then based on these subclusters, we said, okay, overall, the T and NK subcluster correlates negatively with bacterial burden. What of these subclusters does? And you can see that there are some relationships with some of the cytotoxic subclusters, but the one that's most significant is cytotoxic 4, which is a alpha beta CD8 T cells expressing certain cytotoxic molecules. In addition, the stem-like T cells, which I'm not going to really talk about, but they're pretty interesting. A few of these smaller populations, and then the bigger population of the T1, T17, which is this big red population right here. So when we looked at this T1, T17 subcluster, it ends up that, you'd think it might be a TH1, TH17, but it actually is both CD4 and CD8 T cells. And it's classified by a number of transcription factors like BHLE40, which is actually turned out to be pretty interesting in TB, ROR alpha, a little bit of ROR gamma, HIF1 alpha, IL-23 receptor. There's some interferon gamma and TNF, CCR6, CXCR3. Some of these things that look like what have been called in the field and other fields TH1 star or XTH17. And I say XTH17 because we don't see any IL-17 or almost no IL-17 at all in this subcluster, even though we see the transcription factors that would be associated with that. So that we sort of thought of them as XTH17. So given the fact that this is a pretty complicated and big cluster, we subclustered it further into 4 subpopulations, and they're shown here in yellow, red, green, and orange. And orange is a very small, weird population that's just like a lot of DNA damage. And so when you look at what these subclusters look like, they're shown here. So this is the overall T1, T17 subcluster. As I said, it negatively correlates with bacterial burden. This is the subpopulation 1, the red subcluster, and this is the CD4 population primarily. And it kind of looks a little bit like stem-like CD4 T cells. It doesn't have a lot of interferon gamma and TNF, which you can see down here, but it does negatively correlate with bacterial burden. Subpopulation 2 is a CD8 alpha beta, so a T cell subpopulation, and it actually is very cytotoxic, has granzymes, it has perforin, and it negatively correlates with bacterial burden as well. Subpopulation 3, which is this gold cluster, it's both CD4 and CD8, but that's really where the TNF and interferon gamma are showing up in this T1, T17 population. And it doesn't have a very strong correlation at all with bacterial burden, which, you know, everybody knows TNF is important. We all think interferon gamma is important. In this particular study, we did not see either of those cytokines turning out to correlate with bacterial burden. So again, sort of skipping past a ton of data that the paper is on bioRxiv and has been accepted in Immunity. I just wanted to, my computer wants to do weird things. Okay, I just wanted to talk about something that turned out to be sort of interesting. So when we did a correlated abundance, what we asked was, what cell types, what clusters, so we have a number of macrophage clusters, a number of T cell subclusters, which show up most often together in the granulomas. And we ended up with 5 groups. And so group 2 is where the mast cells and the plasma cells in a certain macrophage population and the fibroblasts show up together most often. And then group 3 is the T1, T17s, the cytotoxic 4, another cytotoxic, some of the stem-like T cells. They all sort of group together as well. And then there are a couple other populations. And when we said, okay, how does that relate to bacterial burden? These are the high bacterial burden granulomas, and you can see that they're dominated by this type 2 group of cells that end up together in the granuloma, whereas the low burden granulomas are dominated by the T1, T17, and cytotoxic group. You can see that it varies across granulomas and monkeys, of course, but nonetheless, you can see that as you go to lower bacterial burden, you get an expansion of the T cells, and as you're at higher bacterial burden, you see expansions of group 1 and group 2 primarily. And so Constantine did a really interesting analysis of an interaction analysis of these cell types within granulomas. So on this part right here are the high burden granulomas. And what you can see is the majority of interactions are in this type 2 space. And so if you look at the Circos plot here, the red is type 2, so the group 2. So all these group 2 plasma, mast cells, fibroblasts, they're all talking to each other primarily. And it ends up, we think, being a self-reinforcing, almost like trying to be a wound healing response for those high burden lesions that are all the ones that show up early in infection before there's an adaptive immune response. The ones that show up later in infection have a much more varied interaction across these groups of cells, as you can see in the surface plot, with almost no interaction of the group 2 cells, suggesting again that now we're having really nice activated T cells coming in, and you're starting to really see control of the infection and talking among the group 3 and the group 1 and the group 4, so that there's much more interaction across these different cell populations. So, but one of the things that intrigued me, and the first paper I ever published in TB was about CD8 T cells, was that we saw so many cytotoxic CD8 populations within the granuloma. So we really wanted to sort of hone in on that. So I could have talked about a lot of things in this talk, but I chose to talk about CD8s because I really love them. So what is the role of CD8 lymphocytes in control of TB? So we know that CD4s are important, but what about the CD8s? So some, but not all of the mouse studies suggest a role for CD8s. And I say CD8s because CD8 covers a lot of ground, not just adaptive T cells, but also innate cells. Humans obviously have a CD8 T cell response to TB. Usually people look at interferon gamma, and they don't usually look at cytotoxic responses. In a couple of studies of protection, one that I was involved in, which was giving BCG intravenously, which was incredibly protective in rhesus macaques. BCG IV induces both CD4 and CD8 T cell responses and is really protective. And CMV by Lewis Picker expressing MTB antigens also induces strong CD8 responses and also protects against TB. So we really wanted to take a deeper dive into the CD8s that are in the granuloma. So just to sort of put everybody on the map in terms of CD8s. So CD8, the molecule itself, there's an alpha form and a beta form, and it can exist as a homodimer or as a heterodimer. And broadly, that sort of separates the cells into what we can consider innate-like, if you're CD8 alpha alpha. So this is NK cells, gamma deltas, mates, and NKT cells. And I should point out right here that only about 50% of gamma delta cells are actually CD8 positive. And then there's the adaptive cells. So the classical CD8 alpha beta T cell, restricted by MHC class 1, and then the CD1 restricted T cells, which we're working on with Chechen Sasadri, but I won't be talking about today. So this biology allows us to ask questions about whether or not which types of cells are actually important. And so what we did was we took cynomolgus macaques, is a relatively small study, and we either treated with IgG antibody prior to and during infection as a control, so those are our control monkeys, or we did anti-CD8 alpha depletion, which depletes all of the CD8s, all the CD8 bearing cells, both the innate and the adaptive, because they all have CD8 alpha, or we did anti-CD8 beta, which would deplete primarily the classical CD8 T cells. And so we infected these monkeys with MTB. We did a very short experiment, in part because we didn't have very much money to do it, but also to ask about the very, very early events in TB where the granulomas are being established and starting to grow. And so we just did 6 weeks post-infection. Remember that previous graph I showed you, that's when the bacterial burden in the granulomas is the absolute highest. So we did all of the things we normally do, PET-CT and bacterial burden and flow and single cell, to try and determine if you don't have any CD8s at all, what happens? And if you just don't have classical CD8 T cells, what happens? So just to show you briefly, that in the granulomas and also in the blood and bowel that I'm not showing, the depletion is actually really, really effective. So these are our control granulomas, just showing you what types of cells there are in the granuloma: CD4s, CD8 alpha beta. The red are the double positive CD4 and CD8. We have gamma deltas in orange, 2 flavors, both CD8 positive or CD8 negative. And this is sort of a mixed bag here. As well, we have B cells, of course, as well in the gray. When you deplete the monkeys with CD8 alpha antibody, the granulomas are much less diverse in terms of cell type, so that we actually have the same number of overall lymphocytes in the granulomas, which is interesting, but it's now primarily CD4s, some double negatives, and the gamma deltas that are not CD8 positive, plus of course the B cells. And then when you deplete with CD8 beta antibody, you basically only lose this yellow pie piece, which is the CD8 alpha beta T cells. So it's very effective at depleting just the alpha beta T cells. So just to sort of say, okay, so we have all these cell types, what do they actually do in the granuloma? And we've published previously that actually a lot of the T cells in the granuloma don't do anything at all that we can measure. And I think that probably means that T cells just come to the granuloma because it's a site of inflammation. But in the effector functions that they do have, what you can see, I think, is that if you look at these cytokines, this is CD4 cells, gamma delta cells, and all the different cell types, they don't really make very much in the way of cytokines. So on average, about 10% of the cells in a granuloma, CD4 or CD8, make interferon gamma or TNF or some of those cytokines. In contrast, though, the cytotoxic potential for these granulomas is actually much higher, particularly in the CD8 populations. So the gamma deltas have granzyme B and granulysin, CD8 alpha alphas, which are some probably weird population of innate lymphocytes. They also express a number of these cytotoxic molecules. The alpha beta are strongly granzyme B positive and a little bit of granulysin. And the NK cells are just really positive for these cytotoxic markers. And so we could sort of said, well, wow, there's a lot of cytotoxic potential, but everybody looks at cytokines. And so we said, okay, what happens then if you deplete the CD8s? So this is a complicated graph. Every single line here is a granuloma. They're separated by monkey. So the first the first column are our control granulomas. So these are the percentage of CD8 alpha beta T cells in each granuloma that are making any one of these 4 markers or some combination. So you can see that again, as I showed you, primarily they're making granzyme B. There's a few granulysin in the pink and granzyme B positives, and some interferon gamma here and there. The CD4s are just mostly a little bit of granzyme B and some interferon gamma. They really don't do a whole lot that we've measured. And the gamma deltas are sort of a mixed bag. They like to do a lot of different things, cytotoxicity as well as cytokine production. So then when you deplete all of the CD8s, something really interesting happens. You see an increase in granzyme B in the purple here, production in the CD4 T cells in the granulomas. And you see an increase in cytotoxic markers, granulysin and granzyme, as well as granzyme B in the gamma delta cells. So that was really weird to us. Why, when you deplete the CD8s, do the T cells start to be, the T cells that remain start to be more cytotoxic? And you can see that that is to some extent true when you only deplete the CD8 alpha beta T cells, where you see the gamma deltas start to skew towards a more cytotoxic phenotype as well. So to do a better job of this, because you can only look at so much with flow, we turned to single cell, where we took a number of granulomas from our depleted and control monkeys. And this was done by Sarah Nyquist and Alex Shalick's lab. And again, we have our nice UMAP of all the cell types, and it doesn't look so different than the other one. And we took our T and NK cell cluster and reclustered it. And it turned out in pink are all those cells that would have a cytotoxic phenotype in the granulomas. And this is like from all of the granulomas that we did. And so then we basically named the cytotoxic clusters 1 through 8. And we then looked at them, and it turned out that they kind of pair up so that you have 2 clusters, C1 and C2, for example, that are very similar in their signature. And how they differ is that C1, which is present in the control granulomas, is a CD8 T cell cluster, but C2, which is very similar to C1, is a CD4 cluster. Again, suggesting that somehow the CD4s and the gamma deltas change and become much more like CD8 cells. But what I'll show you is that, in fact, they're like that, but they're not quite as good. And so just to show you this in sort of the UMAP way, the pink is cytotoxic cluster 1. You can see it's present here in the control granulomas, but it's gone pretty much in the depleted granulomas, whereas the orange shows up a little bit in the control granulomas, but is much more here in the depleted granulomas. And so just to give you an example of what that looks like, again, I talked about C1 and C2, and you can see that there are differences in their granzyme productions, even though they have an overall signature the same, and key effector functions like granulysin, perforin, and CCL5, which are only present in the CD8s and not in the CD4s, who are what we call moonlighting as a CD8 cell. The same is true of the CD5 and 6 cluster, which is a little bit more of a gamma delta and CD4 cluster. Again, you see big differences in these major effectors, cytotoxic effectors. And if you score all the cytotoxic effectors between the control, which is always the odd numbers, and the even number pair, you can see that the cytotoxic score is much higher for the CD8s than it is for the moonlighting CD4 or gamma delta. Okay, so why would that be? We have a hypothesis, which is that IL-15, which is a cytokine that induces CD8s, NK cells to be cytotoxic, we think that in a normal granuloma, the CD8s are really responding to the IL-15 that's produced. And we know it's produced by macrophages, monocytes, and interestingly, mast cells. But when you take away the CD8s and you have fewer responders, we think then the CD4s and gamma delta cells become more receptive to IL-15 and start becoming cytotoxic. So the big question is, does that even matter? Are the CD4s good enough to fill in for the CD8s? So this is just sort of a 3D picture of all of the monkeys from the control group, where the orange and yellow means that they sort of the areas of TB disease. These are the CD8 alpha depleted. You can see there's much more TB disease in these monkeys. And the CD8 betas are sort of somewhere in the middle. And you can quantify the total amount of FDG, which is our PET probe, and you can see it's higher in the CD8 alpha depleted group. The number of granulomas is substantially higher in the CD8 alpha depleted group, with just sort of an intermediate phenotype with CD8 beta. There's more pathology, but also more extrapulmonary disease in these CD8 alpha depleted monkeys. So the increased number of granulomas and the increased spread outside of the lung cavity suggests to us more dissemination and therefore leading to more active disease. Now, again, this is only 6 weeks post-infection. Like I said, it's really hard to do worse. So although we see an increase in total bacterial burden in the lungs, it's not very, it's pretty modest, although it is significant. So we wanted to get at this question of dissemination. And so we went to our barcoded strains, which all these monkeys were infected with barcoded strains. So again, every MTB bug that you put in the monkey has a different DNA barcode. We track when the granulomas show up by PET CT, and then we plate each individual granuloma on a separate plate. And then we scrape those colonies individually and make DNA, and then they're sequenced for barcodes. So in doing that, you can make a map of dissemination inside a monkey. And so this is work done by Mike Chow and Forrest Hopkins, as well as Mike Chase and in Sarah Fortune's lab. So just to sort of orient you to the Circos plot, all the orange over here are lung sites, granulomas or lung lobes. Over here in the blue are lymph nodes, and over here in the green are extrapulmonary. And the connections are where we've seen a barcode established in a granuloma and then spread either to a lymph node, spread to an extrapulmonary site, spread to form a new granuloma inside the monkey. And we can do this because we know when the individual granulomas show up. So the red guy spreads quite a bit. The blue guy spreads a little bit here in the lung and then also to the extrapulmonary site. So that's sort of orienting you to this Circos plot. And then this is all of the monkeys. So here's our control monkeys. Again, monkeys are extremely heterogeneous. This guy is a big spreader here. This guy hardly spreads at all. When we did anti-CD8 depletion, I think you can appreciate that there's much more dissemination and spread within the lung to the extrapulmonary space and to the lymph nodes. And you can see this with the anti-CD8 beta to a lesser extent, sort of somewhere in between. So when you quantify that, it turns out there are two things that happen when you don't have any CD8 cells around. One, you get more bacteria established by individual barcodes. We count how many barcodes. So there's some CD8s are forming some sort of block that prevents all the bugs that you put into the airways from getting into the lung parenchyma. And again, that's sort of in the middle here with the CD8 betas. The number of barcodes that disseminate is also much higher, so that you're getting much more dissemination from a number of different barcodes. And the number of tissues sharing a barcode, this is everything from lung to lymph node to extrapulmonary, is significantly higher in the CD8 alpha. So again, CD8s are preventing, to some degree, establishment of infection and also then preventing dissemination of infection. So,
Joanne?
Yep, I'm almost done.
Okay, thanks, because we're running a bit late. Thanks.
It's okay. You know what? I'll just skip this next part because it might take too long. So just in general, I think it's really important to think about the CD8 T cells, and not only the CD8 T cells, but also the innate cells, and to think about cytotoxicity because it is associated with reduced bacterial burden. And we know that the CD8 T cells that are cytotoxic are increased in BCG vaccinated animals. And so enhancing cytotoxicity, either in the context of an adjuvant or a vaccine, is likely to enhance early and later control of infection. So this is, I've mentioned most of my collaborators here, and this is my TB group, and I'm happy to take any questions. Sorry if I ran over.
Great. Okay. Thank you, Joanne. No, it's fascinating. Yeah, unfortunately we have. So I know Erwin had a question that he sent via chat. So Erwin, do you want to?
Well, Marcel tells me I have to get this to go to students first. So please, students, go ahead.
Okay, well, in the interest of time.
Okay, so we are not polite today in the interest of time. No polite. Joanne, thank you. That was fantastic talk. Really, really very impressive. One question that occurred to me, and I'm not sure if you did it or not, when you did the single cell, did you actually do some RNA kinetics in the cell populations to see if there is some maturation of cells happening in the granuloma?
Okay, so that's actually the part that I skipped. It was not single cell. It's actually what we did was we looked at original granulomas over time in different monkeys. And we actually looked at transcription factors. And it turns out that the CD8 and CD4 T cells are there early, but then it isn't till like 10, 12, even later weeks that they're actually expressing things like Tbet and things like that. So I think that is an evolution within the granuloma, although I cannot exclude that they're being recruited later when they're already making that. But we have not done the RNA kinetics experiment. It's a great idea.
Well, that would help, I think. Do you ever sequence, just on the side, when you have these differences, for example, in spreading, do you ever sequence these monkeys?
You know what? We are going to do that. So they're very heterogeneous. And
Yes.
even with the TCR, I mean, Sam Bahar had to go and annotate the whole TCR because these are cynos, not rhesus. But in fact, we think that that's a great idea to sequence them and see if we can make any genetic connection with outcome.
Thank you.
Okay, so I see that both Marcel and Maz have questions.
Can I go with one and then we'll let Maz ask the really difficult question to end?
Please.
Joanne, you mentioned dissemination. For the people who work on human TB, I think we've discussed before, a lot of the articles say that early in the infection, there's a silent bacteremia before the onset of adaptive immunity. I see it very often in review articles. Do you see bacteremia when you're talking about dissemination, or are you talking about within lung dissemination?
Okay, so I don't actually believe that, Marcel. I mean, we have looked, and again, you know, you're sampling a relatively small amount of blood in a big pool of blood. It is very, very rare. And when we do see bacteremia is when a monkey is super sick and immune compromised, SIV, such like that. I'm not convinced that hematogenous spread is the answer to dissemination. To extrapulmonary, maybe, but not within the lung. And so we just don't have any data that that is what's going on. And we just don't see it.
Thanks. Okay, Maz.
Joanne, first of all, I apologize for being 5 minutes late, but
okay, we still like you.
Yeah, it was a brilliant talk. I mean, it's fantastic talk. So I have a couple questions. So the first question that I have, so I guess the killing would be done by macrophages because those are the cells that are infected. So the T cells are communicating with macrophages. So I'm wondering what is happening to those macrophages that actually harboring the bacteria in those granuloma.
Yeah, so are we. You know, first, I just want to say that not all the TB and granulomas, in fact, not most of the TB and granulomas is in macrophages.
Okay.
A lot of it is in the caseum and it's alive. And so
Okay.
you know, you think it shouldn't be. But we have a number of macrophage subsets, and it's very difficult in our single cell to tell which ones are infected and which are not, right? So there's just, in terms of host to bug ratio, the host, you know, wins all the time, and it's hard to do any RNA-seq. So we're not sure exactly. We do have a number of different sort of orthogonal ways of trying to get at this question and try to determine which of the macrophages are actually infected and then which ones might actually be killing. But I think you also have to kill the bugs that are in the caseum, and those could be through secreted effectors that even include granzymes.
Oh, okay. So you think like the production of even some of the cytokine, because there are some literatures like induction of the interferon gamma, for instance, directly can kill bacteria. So you think that that is another mechanism that is involved?
I think both things are probably happening.
Fantastic. So the other fascinating thing that I found out was that you mentioned that IL-15 has the ability to convert some of the CD4 CD8 T cells, sorry, CD4 T cells to become cytotoxic, or gamma delta T cells to become cytotoxic. Can we do that in vitro?
So can we take CD8 T cells, or I mean CD4 T cells, put them in with IL-15 and convert them to the cytotoxic? I think this is incredibly important, and that shows the plasticity of the T cells.
Yeah, so that's one of the things we're doing. There's also a molecule called N803 that is an IL-15 superagonist. So our plan is to actually try and use that as an adjuvant in a vaccine approach, but also just in straight infection to use N803 to try and fire up all the CD8s. Now, whether or not that will fire up the CD4s, we don't know. But I think that in vitro experiment, we definitely want to do that. And we're trying to figure out where's the best place to start.
Yeah.
T cells from the granuloma or what, but that's a great experiment. Okay, I've taken up way too much time. I apologize.
Okay, thanks. Thanks very much, Joanne. Really fascinating talk. I'll get Maz and Marcel to explain some of the finer details to me that I didn't fully grasp. So now again, we're pivoting. So we've gone from kind of global to the granuloma, and now we're going to turn to kind of the human experience. And Madhu is going to introduce Priya Amin, who's going to talk about kind of the TB experience. So Madhu, do you want to?
Thank you, Dick. Priya, are you there? Can you turn on your video? Can anyone see Priya?
I can see her.
Yes. Hi, Priya. Thank you for joining us. Yeah. So nice to have you back at our TB Center. Priya is based in Toronto. She's not just a TB survivor, but she is also a passionate TB advocate. She's part of the TB Conquerors group that Stop TB Canada is trying to put together, and very active within the Canadian TB elimination network. Priya is a seasoned educator in both public, private, and nonprofit sectors, with over 2 decades of experience, from classroom instructions to school management and leadership. She's been an advocate for children and youth for many years, and now she's also spending some of her time advocating for TB as well as mental health. One of her greatest personal achievements has been the completion of her first half marathon. Congrats, Priya.
Thank you.
And she looks forward to completing her first full marathon in the near future. Thank you so much for joining us, and we look forward to your talk on what you intriguingly titled. Go ahead, Priya.
Thank you for that introduction, Dr. Pai. Can you hear me okay, first of all? Can you guys hear me okay?
Yes.
Yes, perfect. Thank you. So when we, as the world first started learning about COVID 2 years ago, I felt a very strange and familiar attachment resurface to my days and months as a TB patient. After all, we were told to social distance, if not isolate, wear masks, and limit social interactions to only members that you live with. Just like COVID, this infectious disease does not discriminate against continents, countries, gender, age, socioeconomic status. Anyone can contract TB. It's treatable and curable as long as you take your treatment. I was born here in Toronto to parents who immigrated to North America from India and Zimbabwe in the early 70s. For most of my life, I had been relatively healthy, nothing more than the seasonal bug from time to time. I work in education, so I had to take the TB skin test on a regular basis over the span of 15 years to satisfy work requirements in the schools. So it was now August 2015, and I was gearing up with excitement to start a new job. I hadn't worked for a year prior to that, so I was really excited to start a new role. But that excitement was squashed when I received a phone call informing me that my test results came back positive for TB infection. With a normal or clear chest x-ray and some symptoms, the infectious disease doctor believed that I had latent TB. A sputum test would later indicate otherwise. I had active pulmonary in my lungs, and later with a CT scan presumed extrapulmonary TB in my lymph node. I didn't know anybody with tuberculosis, nor had I traveled to an endemic country since my last negative test, which was about 2 years prior. Ignorantly, I really didn't know much about TB. In my mind, it was a disease of the past. I was put into quarantine immediately, and a case manager from Toronto Public Health arrived at my house within the hour to walk me through this process. I called my parents and they made their way to my place immediately. The look of sadness and worry and fear on their faces was very evident as they watched my case manager and I discuss the particulars of this disease, standard treatment protocols with DOT program, and the restrictions that would now be placed upon my daily life. From as far back as I can remember, I never wanted to have my parents see me in any kind of pain. My inner child took over, and so I kept this fear that I had at bay. I discovered my awesome poker face, and I laughed more than I wanted to until everybody left. What just rocked my world? I had just completed my first half marathon a year earlier with no training, so my lungs must have been in top shape. I was unemployed for a year prior to my diagnosis with minimal to no income support, and now I would not be able to start my new job for 6 weeks when I was considered no longer infectious. I didn't qualify for short-term disability. I relied on my savings and some support from my parents to get me through. There would be no going to work, earning an income, the grocery store, restaurants, seeing and kissing my loved ones, or social events. I wondered, I'll have to wear a mask if someone visits me. I know it's like run-of-the-mill thing for all of us over the last 2 years, but back then it was super strange to me. When leaving my condo on the 21st floor, I would have to wear a mask to get to my car or simply grab my mail. What would these mandatory medications do to my body? Did I or could I spread this to my young nieces and nephews? And lastly, and most terrifying, could I die? I even prepared a 6-month bucket list, and these thoughts ruminated for many months in my mind. 6 months prior to my diagnosis, I did notice a small lump on the side of my neck. My family doctor concluded that the lump was the result of an environmental allergen and that I was low in iron. Pop some iron pills, he told me, for 3 months and I'll be fine. I went for a second opinion, which returned the same result. More TB education, awareness, and training, in my opinion, is needed for all healthcare workers. The hardest part in sharing my illness with loved ones was seeing and experiencing their fear to be around me, most notably when I was out of quarantine and back to living just like everyone else, but was still on TB meds. From one perspective, I viewed it as a slightly selfish act. And from another perspective, I put myself in their shoes and I wondered what I would do. My sickness was not a well-known one here in Canada, and Google became my best and my worst friend. My relationship with the internet took on a life of its own from day one. Researching the possible side effects of the meds started to create doubt and panic that I would be okay. Soon into treatment, while in quarantine, I learned that my strain of TB was resistant to INH, so it was switched out for another one. I was handling these drugs like a champ with no real limiting side effects. I was no longer infectious, so 6 weeks later, I started my new job. At the end of that first week, I received a phone call telling me that I needed to stop taking my meds immediately, return to quarantine for about 2 weeks as my liver function tests were rising and it was not safe to continue taking the medicine. Unfortunately, I entered quarantine life for another 3 months. I told my boss that I had to keep my stress levels down, so I needed to be off work again as per doctor's orders. I feared losing my job if I told them the real truth. Alarming LFT levels, I was referred for a liver biopsy. The results were benign, but those meds wreaked havoc on my insides. I was admitted to the hospital for a few nights on an NAC drip to lower my LFTs. which was successful. The option of going on amikacin was discussed, as that would get me back to work and out of quarantine. However, with the potential side effects of losing my hearing, I opted to cancel my PICC line insertion 2 times. One of the physicians at the clinic was not happy with my decision, as he was concerned with the reputation of the clinic with the big hospital that I had scheduled to have my PICC line insertion at. Good news arrived later at the end of the year, just in time for Christmas. My LFTs were trending downwards, and I started a new drug regimen in January of 2016 and returned to work and regular life at the end of that month. I completed the full treatment of 9 months of medication. Popping those pills before work almost always led to vomiting, nausea, some joint pain, and fatigue. There were many days where I didn't want to take my medicine, but I reminded myself of the importance of doing so and didn't take for granted that living in Canada meant that I had access to treatment. I was officially discharged as a TB patient in October of 2017 and was invited to share my story for the first time in a room full of health science students at U of T, academics, TB health workers, outreach workers, my family, and some friends. I didn't realize the power of my own voice in the TB community until that moment. It's important for TB survivors to rise up, to share your story, to advocate and support others living with TB. It is a long and difficult road. Thankfully, contact tracing produced no other infections in my network of people. And my dad, who was born in India, who wasn't sure if he had the BCG vaccination in India as a baby, stubbornly agreed to my younger sisters and the infectious disease doctor's plea to take rifampin for 4 months for possible latent TB. After all, like many other retired South Asians in our community, he would travel to India for a few months every winter for a vacation. My dad initially didn't want to take the medicine, as he said, "I can't live like you had to live, Priya, in quarantine. If something happened to me, who would take care of Mom?" That hurt me, but I was really good at wearing this mask and this armor for everyone. During my sickness, while I felt very alone and isolated and depleted on some levels, the relationships that I formed with my TB nurse and case manager were instrumental in my experience. I would remind them on a regular basis that I wanted to become a volunteer with Toronto Public Health to raise awareness and advocate for other patients. We would laugh together. They would share stories of other TB patients, respecting anonymity, bring me a few food items that were high in protein value, and would most importantly, actively listen without judgment. Admittedly, though, I put on a much braver face with them too. I would learn about the dire experiences and diagnosis of others in the province. I would learn about the dire experiences and diagnosis of others in the province, the country, to the north, on the reserves, and around the globe, and could not imagine what their plight would feel like for me. I had a complicated case, but not a super aggressive one. I'll never forget the time I was waiting to pick up my meds from the pharmacy at the clinic when a young lady approached me and said, "Do you have tuberculosis too? Is this your first time picking up your medications?" In that moment, I was compelled to keep her spirits as high as possible because I remember being her, my first time, and how daunting it was. And as such, I replied, "No, it's not my first time. I've been on meds for about 6 months and you're gonna be okay. You may feel a little nauseous and a little tired after you take your meds, but you'll keep going." She said that she was 21 years old, had just moved here from Africa, had a 1-year-old at home, and would have to take medicine for 9 months. She had latent TB. Her distress and fear were evident as she wondered if she could handle it all. I replied to her, "Yes, you can and you will. All is temporary and this too will pass." As a TB patient, the ability to connect with TB patients, past or present, is vital to the journey that one goes through. Reading case stories, mainly online from those in other countries, is eye-opening and provides some solace. However, the need for live contact in person, via phone or digitally, in my opinion, has the capacity to ease the hardships of having TB. The physical effects are one thing, but the social, emotional, and mental toll are another. COVID has leveraged digital technology in almost every sector and has allowed us to stay connected to our people and work in a way like never before. It's been freeing this past week to be out and about without having to wear a mask. But the intangible mask that I wore during and after my TB illness shielded my loved ones and colleagues from my discomfort, my worry, my fear, my lack of motivation, and this notion that coexisting in the world worked for me. When I did take off this mask, I was met with some resistance to be around me for fear of catching what I had. A breakdown in trust and reliability in some relationships that I had in my life for years occurred. The days of the pandemic allowed me ample time to reflect and reevaluate my life since my diagnosis. I realized that parts of me suppressed what needed attention, processing and healing if the goal of truly being present and living my best life was to be achieved. In many ways, I missed the pre-TB me. If you were present for any of the mental health therapy sessions I've had over the last few years, you would quickly realize that I was carrying a lot of unhealed, unaddressed TB baggage. Currently, I'm amidst what I call PREA version 3.0, and this version finds me reclaiming my physical, mental, and emotional strength and coming more into spiritual alignment. If I was diagnosed with TB today, for the first time, I would ask for help. I would tell my people what I needed from them. I would seek out professional mental health support during treatment, nutritional counseling, and filling my own cup first as much as I could versus someone else's. Ask more TB questions to the TB experts, definitely ones pertaining to post-TB care around social, emotional, and mental health during treatment. I would tell anyone going through TB treatment to do the same. My message to you is that you will be okay. You will start feeling better as long as you stick to the meds and try to be less concerned about where you got it from. Don't be afraid of the word surveillance if you come across it in any of the literature. It simply means that you'll be monitored by TB health workers and experts to ensure that you take your meds and fully recover. Also, ask your healthcare team to talk to you about what you can expect during and post TB. Push the narrative that people don't have to fear being around you, especially when you are no longer infectious. My first time at clinic, when I asked the doctor what my options were if I didn't want to take the medication, because taking Advil was a big deal for me, he said, "Priya, in this country, if you don't want to take the medication, you can sit at home alone until you die. You are a threat to the public if you don't take the medicine. While I understand the intended message that without treatment you can die, I would have preferred there to be some more bedside manner at play." It was very harsh to hear. It was also alarming to hear that I shouldn't tell people at work that I have TB because they will not want to be around me. They'll gossip, etc. And I'm starting a new job. I'd already told my boss. How else could I explain now not showing up for my new job? While my diagnosis was leaked to staff during my absence in quarantine, when I was confronted with it, I chose to assure people that I was okay, that I'm not infectious, and educate them on this disease. During that chapter of my life, which was filled with denial and despair, uncertainty and fear, it was gratitude and strength, unwavering support from a cherished few, acceptance, and those meds, along with a lot of humor, which is what I believe propelled me forward to rise above and conquer tuberculosis. To this day, when I have a dry cough or I'm unusually fatigued and I lose weight inexplicably, my mind is met with some paranoia that TB has returned. I have tested 3 times since I was discharged, the last time being in August of 2020. I've done more COVID tests than TB tests, and I'm glad that both tests, both kinds, have been negative. Unfortunately, a deep-rooted awareness remains in me that I could get it again. However, if that's what fate has in store for me, I'm much better equipped to conquer it in an authentic, maskless way. So in closing, as a member of the Stop TB Canada Steering Committee, I remain motivated to learn and become more involved with the provincial, national, and global initiatives to end TB, while highlighting the need for improved patient-centered care. I'm happy to share that over the last year, an initiative to support Canadians with TB has been created by Stop TB Canada. So I ask all of you to please share and encourage anyone that you know that has had or is going through TB treatment in Canada to join the TB Conquerors Facebook group. It's a safe and protected space to share, support, educate, and inform. This is not another world's problem. It's within our own national borders, provincial and territorial. No one should feel alone in this treatable and curable disease. COVID has taught us many things. One thing is what's possible when people come together on a human level. And another notable thing that COVID has taught us is what is possible when money and resources are thrown at an infectious disease. We were able to have a vaccine available in a year. How is it possible to work so quickly on a vaccine for COVID-19, which we are really grateful for, but not for a disease as old as TB? Can't we do the same thing for TB? Thank you for inviting me here today to share my story. It means more than you know.
Thank you, Priya. We are very, very grateful to you. We can see how just recollecting the story is so stressful and traumatic to you. And we know you've done it before and you continue to do this because I think we all need to hear it and be educated. So you're taking on that burden for us. So we're very grateful. And really exciting to see that there will be a network of TB survivors in Canada who can support new people with TB within the country. Robin, would you want to say anything about how we could all tap into the resources, our clinicians, for example?
Yeah, of course. Thanks, Madhu, and thanks, Priya. So great to see everybody here virtual today. I shared a link in the chat to the Stop TB Canada webpage for the TB Conquerors group. That's where at any given moment or time you can go and see the latest information and get engaged with the TB Conquerors group. If you're engaging with people affected by TB, our biggest ask of you is to direct them to the TB Conquerors group. Tell them that there is a Facebook group that they can join. Tell them that they can reach out to Noang, who is really leading and developing this network right now. If they need any support, that there are ways to engage with people who have lived through surviving and conquering TB. So check it out there. If you work in a clinic, there is a poster on that webpage. You can print that poster out and put it up in your clinic. That's another really easy thing you can do to help us build this network. And follow us on all the socials, the Instagram, the Twitter, and direct people in our directions, and get involved more broadly, I'd say, with Stop TB Canada. So right now, Stop TB Canada is a purely volunteer led initiative. It's people who care about ending TB at home and abroad that are putting in time off the corner of their very already well-rounded desks to try and take collaborative, coordinated advocacy action to end TB at home and abroad. And it's working. We are having an impact already. This network's been reinvigorated just in 2 years, and we are making lots of noise and making change. One I can say I'm really proud of today, we engaged the Minister of International Development, the Honorable Hajit Sajan, for the first time ever at a World TB Day event here in Canada. So we have the political attention. The power of us working together as a network is working. So engage with Stop TB Canada more broadly too, and support us. If you have some extra time, you want to chip in and give some in-kind contributions or even some dollar bills, that would be helpful too.
And so is it correct that Canada today announced $11 million for TB Reach?
Yes, $11 million for TB Reach. Yeah.
And we are also continuing to contribute to the Global Fund?
Yes, so Canada remains an investor in the Global Fund right now. And I know Stop TB Canada, Results Canada, lots of other allies are going to be campaigning really hard throughout this year to make sure that Canada steps up with an ambitious investment in the Global Fund later this year as well.
And I think, Dick,
Great.
the other internal
Thanks.
advocacy would be to push Health Canada to update our TB report, which was last published in 2017. It's a disgrace to not even have updated TB data for 5 years. I mean, what is this?
Okay, well, sure. But before I comment on that, again, have a comment or a question for Priya? Marcel?
Marcel?
Hi, Priya. That was really wonderful. Thank you for presenting. I'm following up a bit on what Madhu was saying, but I'd like to have your voice in this. As you may have read in the Globe and Mail a week ago, there haven't been Nunavut TB stats available, and it's thought to be due to privacy reasons. Canada doesn't have a TB report recently. How do you feel about having the data out there? And is there potential for harm? And how can we do it? What will be the best practices?
When you say potential for harm, what can you expand on what you mean by that?
Well, I think the idea is, as I understand, if Gonzalo is on, maybe he can explain more, that if somebody identifies that there were 3 people with TB in a village of 300, and you know that 3 people in that village of 300 went south to a hospital for a month, you may be able to say, oh, therefore those are the 3 people who had TB. On the other hand, we risk stigmatizing the disease by not talking about it. So that's why I want to hear your perspective on whether we should be running with TB data the way we have COVID data, and it's up on the CBC website every week, and we know the information so that we can work with it, or if there's something about TB that requires us to suppress the information.
Yeah, thank you for clarifying that. I am a strong believer, and I don't understand why we're not publishing that data and why we don't have access to that in the same way that we have had for COVID. I think it's crucial if we want to meet our target and our goal of ending TB, especially in the North by 2030, and then globally. I don't understand why we can't be publishing it. I think it's harmful to not publish it.
Thank you for your perspective.
Okay, so maybe that's a good segue to the next speaker. And I have the pleasure of introducing Gonzalo, but before I do, I want to actually show something myself, if I may. And that's, of course, as usual, if I can find it. So that is the Canadian TB standards are just being released. Officially, they're released today. I was supposed to have a link, but in fact, I'm not quite sure that they're actually going to be released today, but in any event, they're going to be released, within the next 24 hours, let's say. And we're still going to call it that it was released today. This is the 8th edition of the Canadian TB standards. And again, the next speaker, Gonzalo, is going to talk in detail about one important part of that, one chapter. But I just wanted to take a moment to mention that, this World TB Day, the Canadian TB standards are officially out. They will be online at the Canadian Journal of Respiratory Critical Care and Sleep Medicine, and I will be happy to send the link to everyone who joined this call today as soon as I have it. There you have the editors, and here is a long list of the authors, many of whom are on this call today, I could say. And if you want to see them, we had an all authors meeting by Zoom. And here's some of the people who are contributing in different screenshots, all of us looking a bit Zoom-like. This was towards the end of the meeting after 2 full days on Zoom. So the Canadian TB standards, just to mention for those who don't know it, it's intended as a practical guide for frontline providers. It's kind of a common observation of many physicians, nurses, and other healthcare providers that you don't get much about TB in your medical school or your nursing school because, frankly, you don't see it that much in urban centers. I mean, Priya's, I think, kind of attests to some of the problems of knowledge. And so this is intended for people who've maybe not heard much about TB, but are going to somewhere where they're, or are caring for a population where there is TB. This is the 8th edition. It's heavily revised from the 7th, which was published in 2013. It's evidence-based. I would say the evidence was updated by the authors up to December. So already it begins to become out of date, like all sort of textbooks. It has basics on epi and pathogenesis, clinical aspects, and then public health aspects. And really the last slide I'm going to show, because everybody was mentioning this. So in this, we do have, in chapter 1, we do have the latest information on TB. This is only one of many slides or figures and tables. There's a lot of detail on the epidemiology, including in different populations and different parts of Canada. One thing I would note, a couple of things. First of all, really in the last few years, the numbers of individuals diagnosed with TB, previously termed cases, has actually increased, and in 2019 was higher than at any previous time across Canada in terms of numbers of individuals diagnosed with active TB. The rate is kind of looks about the same. 2020, we did see a drop in the number of individuals, but I think this reflects what Dennis was talking about at the beginning, which is underdiagnosis, and that there will be, unfortunately, perhaps a catch-up, so to speak, in 2021. So I'm going to stop that because this was sort of just to really announce the Canadian TB standards. And again, we'll share the link with everyone as soon as we have it. But it is, or you can also go to the website of the Canadian Thoracic Society, and you should be able to find that link shortly. So back to the real agenda that I stole for a moment there. So Gonzalo is very respected associate professor at the University of Ottawa, scientist at the Ottawa Hospital Research Institute, head of the Division of Respirology. I can sympathize with him over that one. And he has worked for many, many years in Nunavut, and along with Inuit partners, he founded Taima TB, which for those who don't know means Stop TB in Inuktitut. And he founded that back in 2010. And again, it's kind of part of the sort of the strengths and of Gonzalo is that it's still running strong. So Gonzalo, over to you to tell us a little bit about the developing, writing, the experience of engaging the Indigenous perspectives in the new chapter on Indigenous tuberculosis and in the new Canadian tuberculosis standards. Thanks, Gonzalo.
Thanks very much, Dick. Can you hear me?
I can hear you.
And can you see the presentation?
I can see it. It's in whatever mode. Yeah, now it's in perfect.
All right. Well, thank you very much, Dick, for inviting me to talk at the TB Research McGill. And I also wanted to congratulate Priya for that really fantastic presentation. It really reminds us why we do everything that we do. And hopefully this will segue kind of nicely from her talk. So what Dick asked me to talk about is basically we were tasked with putting together the Indigenous people of Canada TB standards, and what we wanted to talk about is working with researchers to develop the guidelines. So I'm actually not going to talk about the chapter itself as much, but more about the process and how we did it. So I'll start off with a land acknowledgement. We pay respect to the Algonquin people who are the traditional guardians of this land, specifically the Ottawa region where I am dialing in from. We acknowledge their longstanding relationship with this territory, which remains unceded. We pay respect to all Indigenous people from all nations across Canada. We acknowledge the traditional knowledge keepers, both young and old, and we honor their courageous leaders, past, present, and future. Just a couple of disclosures. I've received funding from the Public Health Agency of Canada and CIHR. And for this presentation in particular, I wanted to make some personal disclosures. I think first and foremost, I am not an Indigenous person. In fact, I was born in Santiago, Chile, and I came to Canada, I always say as an immigrant/refugee after a military coup that happened in the 70s. And that's how I made it to Canada. And I am forever grateful to Canada and continue to try to contribute to this country for allowing me to come here. I've also been working in Nunavut as a respirologist and TB consultant since 2007, as Dick mentioned. I also was part of, along with my Inuit partners, we established this Time of TB research group that's been going for about 10 years. So I just wanted to give you a little bit of background as to who I am and what angle I'm coming from, and to set the stage in terms of what we wanted to talk about today. So, the idea here is to highlight some of the lessons learned in putting together the Canadian TB standards for healthcare workers serving Indigenous peoples of Canada. Again, I want to make it clear this is from a non-Indigenous TB clinician/researcher perspective. So I'm hoping that what I can do is transfer the lessons learned to the researchers that are on this call that are going to be working with Indigenous peoples in the field of tuberculosis. So just wanted to highlight that the purpose is not to review the article content. And in fact, Jonathan Dunn, one of the members of our committee, is going to be presenting this at the Canadian Respiratory Conference in Victoria. I believe it's 1 or 2 weeks. And he's also going to be presenting it at ISC. So we'll send this group those links to be able to hear Jonathan's presentation, which is actually excellent. I've already had a chance to hear it. So again, we're going to highlight the processes and principles used to produce this guideline. So a little bit of background first. Indigenous research, I think for non-Indigenous researchers, we have to remember that we have to come from a place of humility and not from a place where we know it all. And I think that that's a little bit of a contradiction in of itself, because as TB researchers, often people look to us because we're supposed to be the experts. And so sometimes right there, that's what kind of one of the first messages that I'll send to you is that we may be experts in our field, but we are definitely not experts in Indigenous issues. And so I think it's really important, again, to come at this from a place of humility and remember that we are experts in our field, but we are not experts in Indigenous issues. And that in that back and forth, we also have to respect Indigenous worldviews and how they pertain to TB research. And I think that's also a really key concept that I want you to think about as I take you through this process. So where did this all start off? Well, it's incredible, but it's now 10 years since I think way back in 2012, I was tasked with doing a systematic review of all things TB in Indigenous populations for the 7th edition of the TB standards. So I took that, as marching orders to go ahead and do a systematic review on the topic, which we did with a committee of TB experts in the field. I think that it did meet what was asked in terms of the review provided readers with a starting point on the topic. But one thing I think that we all felt that it missed was that it really didn't ask the question, what are Indigenous peoples interested in knowing about TB? So that's really kind of the starting point of where we started when we developed these 8th edition of the TB standards. So we decided to take a different approach And the concept that we used was nothing about us without us, which is actually a concept that's been used in many different disciplines, but in the Indigenous research arena, certainly this is an important concept. And of course, it's very explicit in nature, in that you have to involve Indigenous peoples in work that's being done, especially when it's about them. And so the first step that we decided to take was by asking each of the 3 Indigenous groups of Canada to appoint a representative to the guideline committee. So from there, we said, so there are 3 Indigenous groups in Canada, in particular First Nations, Métis, and Inuit. And so when we first put together the group, we decided that we would have Indigenous representation, but we would also link each Indigenous representative with a clinician that was going to be in a supportive role, and more on that in a second. So we started off by making it clear that the Indigenous representatives were going to tell us, the TB clinicians, what they wanted from the guidelines. And here are a couple of quotes from that original meeting of the minds, as they say. Even if you have all the latest medical studies, if you do not know the context of the people you are serving, then it becomes very challenging to implement the new medical discoveries. TB care does not stop after the prescription is written. There is a lot more to TB among Indigenous Canadians than just the medical studies. The context, specifically the history, culture, and epidemiology of each Indigenous group, is central to being able to understand and care for individuals with TB in these regions of Canada. And so, I mean, these are just quotes that summarize the discussion that we had, but it certainly set the tone for the ongoing meetings that we were going to have, and what was going to develop into the guidelines that we were going to work on together. So we decided that we also wanted to kind of establish a little bit of important governance issues, and we made decisions, and we all agreed to work by consensus. And in fact, all 3 groups have very interesting models of consensus. For example, First Nations have a talking circle model. Inuit have a consensus model as well as there is a Métis model of consensus. Now, there are people who are much more learned than I in what consensus means within these 3 groups in Canada. I would really encourage all of you to further learn about this. But essentially, it really moves away from the classic hierarchical structure. We remove the hierarchical structure, and we make decisions together. And what it really also means is that we sit around a round table where we are equals, seeing eye to eye, and sharing stories. And to be clear, throughout the time that we were working, there has to be, and there's going to be, disagreements. Those disagreements are then worked over by further discussions, and also asking others in the committee to weigh in. And of course, in this whole process, we learn, which is a universal trait, I'll say, but listening is paramount. So obviously that is key. And there is literature actually on these models, if anyone is interested. And specifically, I know more about the Inuit consensus models, and there is a publication on this in healthcare, which I thought was quite interesting, that we applied. The downside to consensus is that consensus takes time, and most of us researchers are very low on time. And that is definitely an issue, because it does sometimes clash. But my committee members and Indigenous representatives really helped me understand that this unhurried meeting style really is necessary, because it attempts to listen to everyone around the table, and you need time for that. I'll tell you that I think we probably broke the record in terms of the number of meetings that we had in order to produce these guidelines. There was over 20 meetings that we had. The meetings took about 2 to 3 hours at the very least. In fact, the first meeting we had was December 2019. Now, of course, there was a pandemic in the middle of all this, but nonetheless, it's just to kind of give you an idea that you have to budget more time in working with Indigenous communities, again, because I think the best thing to do is to adopt this unhurried meeting style and take your time, because it really does help. I think someone might have their microphone on. I'm not sure, but if you guys could put it on mute, that would be great. So the other thing I'll say is, that's really important is that language matters. And going back to the concept that I initially introduced, in saying that we really must respect worldviews, Indigenous worldviews. I think that often, there are miscommunications that occur, mostly because, and often from differing worldviews. And I think that one of the things that I did, even though it took me probably about an hour after every meeting, I would personally write the minutes to every single, most of the key meetings that we had. And interestingly, most of the time when I would do that in other meetings that I host or this, I've never usually had anything edited or anything like that. But in fact, often, in those meetings, I did have the concepts that we put forward edited or at least changed, and it allowed us to again get back to this issue, this idea of consensus, and being able to go back and say, okay, you said this, but actually we understood this, and getting to the crux of the ideas, and really dissecting language and ensuring that we were on the same path at every step, and that we didn't diverge early on in the path, right? That we continued to stay on the path. And I think that also was a key learning point, and something that I would pass along to this audience. The other thing that I would say, working with Indigenous communities or Indigenous organizations, is that typically you'll have Indigenous representatives that come to the meetings, but it's important to note that they represent an organization and that they must get organizational approvals, and those approvals take time, and they take 2-3 months sometimes. And what needs to be understood is that these organizations are being they're being asked to do so many things from all walks of not just in health or science, but all kinds of policy work that they do and everything like that. So they have obviously few people to do this, and so obviously these things can sometimes take time. So an important concept to remember when working with these groups. I think also very important, I mentioned that there are 3 groups: First Nations, Inuit, Métis, 3 official groups under the Canadian Constitution. But we must always remember and respect that these are distinct groups within Indigenous peoples of Canada, and they have separate cultures, geography, and history. In fact, First Nations peoples, for example, have over 600 bands across Canada. And I mean, it's just important to understand the rich diversity, and it's really incredible, an incredible thing to study. But what one has to remember is that they are distinct. And my committee members reminded me several times of this. And there's no doubt that the groups commonly experience the processes of colonization, and they have several other areas of overlap, but again, their cultures are distinct, and that's how we should do our work and do our research. But one of the things that we learned and that came out of developing this guideline was that at the same time, we learned that their TB histories did have similar parallels, whether it be getting diagnosed with tuberculosis in remote areas and having to come to urban areas in order to get treatment, and sometimes for years at a time, leaving communities, resulting in transgenerational emotional scarring. We heard that loud and clear from all three groups. We also heard, for example, the impacts of residential schools and the enormity of the number of cases of tuberculosis that erupted in those schools. These are just to give you some examples of the things that were brought up in our discussion. So like I said, the concept that I'm putting forward here is that although they definitely, each Indigenous group are distinct, we did in this guideline find some parallels within their TB histories. So where did that kind of leave us? We said, okay, we really worked on what the purpose of the document was going to be. In fact, we probably spent the first 6 months just debating that issue in terms of what we wanted to tell people on the front line and how and what these guidelines were going to do. And this is what we came up with. So the purpose of the document is to provide healthcare workers going to Indigenous lands to do TB work with an introductory platform to learn about the specific epidemiology, historical, and cultural context for each of the three groups in Canada in an effort to improve cultural competence with respect to providing TB care for Indigenous peoples in Canada. So the committee agreed to have each Indigenous group provide a specific section according to a set outline that we had all agreed by, obviously by consensus. And I think that was important again, because we were trying to balance the issues around distinct groups, but also trying to work in the ideas of some parallels in what has occurred in terms of Canadian TB history as it relates to Indigenous peoples of Canada. So the other thing that we had done, the other concept that we did here was we kind of turned the tables in that we said, look, instead of the clinicians, writing these guidelines, we wanted our Indigenous representatives to actually take the pen, have the pen, and write the guidelines. And us and the TB clinicians, we were going to be mostly in a supportive role in order to support, expand, and find evidence as well, to help find evidence for the points of view of the Indigenous representatives. And we wanted to highlight the oral traditions of Indigenous peoples and lived experience by finding video testimonials to support the statements made. So that also kind of like a different concept. We wanted to use the gray literature. So we really dove into that, which is very different than our comfort zone of looking for published, published articles to support positions. And so we really tried to do that as much as possible, because all three Indigenous groups certainly do work within oral traditions more than written traditions. So one of the other things that we did, another kind of concept that we added, is that we wanted the leaders of each of the groups to provide a statement. And I encourage all of you to read the statements. They're quite interesting in that they're quite different, but at the same time, you can see weaving of the key themes that do find parallels between the three of them. And so I wanted to thank National Chief Roseanne Archibald, National Chief of the Assembly of First Nations. I also wanted to thank President Natan Obed from Inuit Tapiri Kanatami, and also Cassidy Caron, the President of the Métis National Council. So again, I thought it was really a great feature to be able to have the leaders talk about tuberculosis and what it means to them and what they see into the future and things that should be done. So our final product was basically this chapter, which we hope by the end of the day, or as Dick said, within the next 24 hours, will be up. I won't go through the 7 points that we generated, because what I'd like you to do actually is to connect when Jonathan Dunn, who is Ojibwe himself, presents this at the Canadian Respiratory Conference, but also we will have him present it at Indigenous Services Canada. We'll have a webinar that he will do that. So I encourage you guys to come and see him do the real presentation, I'll call it. So this is my second last slide or so. I'll just say that whenever I write a paper or whenever I write guidelines or anything like that, I always like to reflect back and say, well, what could I have done better? What were the limitations? I mean, there's always room for improvement. I think the first thing that I'll say is that, I really wanted to do a better job of highlighting the voices of indigenous peoples. And I think this segues very nicely with Priya's talk in that I really think, in all of these guidelines, in all of these things that we put together, putting the patient first and having actual TB survivors speak exactly like Priya did, really has a real impact on everybody who listens to that. Not only inspires, but it also reminds us why we do what we do. And it also is able to give us a better understanding of what's happening. And of course, lived experience is of paramount importance in this domain. I think we could have done a better job of that. I think that we definitely have links that are there. It would have been nicer to have a better way to highlight them, but I encourage all of you to go to the references and click on those videos, because you'll actually hear real people giving their impressions on either surviving tuberculosis or having lived experience in indigenous lands. So we did try to use these video references. Again, may not have been as clear as I wanted. Also, there were several points brought up about the fact that, we talk about cultural competence in this document, which is very different than cultural safety, and it's important to note that difference. And a shout out to Dr. Falzon, who really made us think about this point, and he is very well correct in saying that really all we did in this guideline is make an introductory statement. Our only way of trying to answer this was to say, okay, you know what, we're going to add a whole bunch of websites that were approved by our indigenous partners at the end of the document, so that you can actually go and get training, better training, and specific training to where you're going to go. So I think that's it. I wanted to thank the committee members that had an incredible amount of patience with me, and certainly gave me a lot of space to move around and try different things. And they taught me a considerable amount of time during the production of this guideline. We hope that this guideline will assist healthcare workers and public health professionals, that is, to serve Indigenous peoples of Canada in TB care. And I want to thank Jonathan Dunn, Marlene Larocque, Eduardo Vides, Deborah Van Dyke, Tom Wong, Faz Khan, and Richard Long for helping put together these guidelines. Thank you.
Thanks very much, Gonzalo. Great talk. Really quite a process, as you say, with indeed probably more hours of meetings than most of us put into writing and other form and evidence review for the chapters. So a tremendous commitment there to communication, to listening, communicating, and so on. So yes, I look forward, I'm sure everyone looks forward to reading the new chapter. I just mentioned there was a question in the chat about where would they find the link. So it's the Canadian Journal of Respiratory and Critical Care Medicine, and that's quite a mouthful, but hopefully the Canadian Thoracic Society, I think if you go to their website later today or tomorrow, they should have something noticed there. Okay, so now opening it to questions. Amrita, did you want to say anything, or you're there, I know, so.
Hi, Dick. Hi, everyone. No, I just wanted to say that was awesome. It was so nice to actually hear the process of the guideline development as opposed to the guidelines itself. Thank you.
Thanks, Amrita.
Mm-hmm.
I might comment, Dick, because Marcel kind of floated the idea about the issue of data and data reporting. I think it's a good point you bring up, Marcel. It's a very hot topic. The journalist who wrote that piece basically did put in a formal complaint to the Privacy Information Officer of Nunavut. And I don't know if you saw that, but the Privacy Information Officer sided with the journalist, not the government. But what you do learn from this is that the government even though the privacy commissioner can point these things out, it doesn't really have teeth in that it can't compel the government to release that data. I have to say is that one can't ignore the fact that for the COVID pandemic, I mean, they've been publishing weekly levels of COVID data from each of the communities, and there hasn't been any problem with that, at least that we know of. So it's a hard debate. It's a hard debate. I think it's going to be continue to be debated, but I think that the government of Nunavut still doesn't feel that that it should be released. But I think that might be changing, and I hope so. In that, I think that it does help. I mean, it does help to know where cases are coming from, and we've learned that so acutely with the COVID pandemic after these during these last 2 years, and I hope that's the way it's going to go. But we'll have to wait and see.
Okay, so Marcel's nicely asked if he has permission to ask a question. So
there you go.
Please.
So Gonzalo, rather than knowing what the government of Nunavut wants to do or not, what is the opinion of the ITK, the opinion of the Assembly of First Nations, and the Assembly and the Métis leadership? And it needn't be the same, but since you're working with all three, I would be very interested to hear their perspective.
Yeah, no, that's a good question. I mean, it wasn't part of what we were doing with the guidelines, but certainly a question that we could, present to them. I think typically what folks are always, and it speaks to who speaks for a community, right? It really speaks to that. You may have, there's 25 communities in Nunavut. You may have 20 of them that think, yeah, just publish it. It's go ahead. But maybe there's a couple that don't, and then how do you meet there, right? And for example, for Inuit, it would be a consensus that would have to come, and so there would have to be more work done on that. We're currently actually working to publish one of our papers, and it was of a community-wide screen in a community, and what we're going to do and what we are doing right now is we're in talks with the hamlet itself, and we're going to ask them, what would you like us to do? And then we're going to do what they tell us. But it would be nice to have a national level kind of, consensus on that. But I don't think we have that. And I don't know whether the government's going to want to kind of, stick their neck out when there may not be complete consensus. That's my take on the issue.
Thanks, Gonzalo.
Okay. Thank you, Gonzalo. Listen, we're, as always, running a bit behind, and with Zoom, you have to be right on time, smuck on time, or people are Zoomed somewhere else. So we're going to carry on. So I just mentioned, so we now have 3 oral presentations. I'm sorry for those of you. We planned this with a very tight schedule and really no breaks. Which perhaps was an oversight, I realize. In any event, we're going on to the oral presentations. I'd like first to acknowledge the judges who judged the abstracts that were selected out of all those submitted. They picked the best 3 for oral presentations. So that was Olivia Oxlade, Faz Ahmad Khan, and Simon Grandjean-Lapierre, who between them picked these 3. The judges today will be Marcel Baer, Erwin Schur, and Madhu Pai. And a note to the 3 judges, please send us actually to Lysandra or Judith, your ratings as soon as you can after the last presentation, so we can then compile the scores and announce the winners before the close of this research day. So, also just kind of the rules of engagement, so to speak. Each trainee has 12 minutes to present. That's it, that's all. Although I don't have a mute function since I'm not the host, as I just discovered not long ago. After that, there will be 5 minutes for questions, more or less. And again, the best is to send your question through the chat, but try to do it just at the end, since sometimes presenters find it a bit distracting when the questions pop up. And either I can read the questions or I'll call on you to read the questions. Judges also may have questions. They just need to let me know. Okay, so the first presentation is by Aria Ed Jordan, who I see is all set, and is entitled Tuberculosis Infection Prevalence Among Foreign-Born Canadian Residents in 2016. Go ahead. And sorry, just another note, please, everyone mute yourself, except of course for the presenter.
All right, everyone can see my screen, I'm assuming. All right, so hello everyone, thanks for having me here. My name is Aria Jordan, and today I'll be presenting tuberculosis infection prevalence among foreign-born Canadian residents in 2016. So to begin, in Canada, so Canada is a country with low TB disease incidence, and progress toward TB elimination has stalled. We expect this to become more difficult as COVID-19 begins to present another challenge. Looking on the right, we see the stagnation in TB disease incidence in Canada, and we see that incidence has remained higher than that of the United States, despite Canada providing wider access to care generally. So socio-demographic shifts have occurred over time in Canada, such that TB disease now disproportionately impacts vulnerable groups such as migrants. The stagnation in TB disease incidence in Canada can largely be attributed to untreated TB infection that progressed to TB disease among these vulnerable groups. And as a result of this going unaddressed, approximately 70% of foreign-born in Canada have TB disease, despite only comprising 22% of the population. So approximately 85% of TB disease among foreign-born persons reflects progression of TB infection that progressed to TB disease prior to arrival in Canada. And for this reason, reducing TB disease incidence will require greater focus on TBI screening and treatment measures. So to better tailor prevention and care efforts, we need to know more about the picture of TBI in Canada. However, prevalence of TBI in Canada is unknown. So to overcome this and inform TB elimination strategies, we estimated TBI prevalence among foreign-born persons in Canada for the year 2016. To do this, we extracted 2016 Canadian census data. The data was organized by place of birth, age group, and year of arrival group. Our data was split, so we had to distribute the population evenly within each group to break down age and year of arrival by individual year. So we also imputed year of arrival prior to 1981 to as far back as 1931 according to age group. And in the end, we had a data set that comprised 7.2 million or 96% of foreign-born persons in Canada. For our model, we built upon prior global age-specific estimates of TBI. These had been generated by our colleagues in the UK, Ryan Hoban and Pete Dodd. So they had actually estimated global tuberculosis prevalence back in 2014. So building upon their previous work, we simulated 200 annual risk of infection trajectories to generate historic estimates of yearly force of infection for 217 countries. The annual risk of infection estimates were based on TST surveys, and where TST surveys weren't available, a revised Stabilo ratio was used, and the Stabilo ratio used accounted for uncertainty by relating smear positive TB disease to annual risk of infection. We summed the FOIs for each year of residency in the country of birth before migration, as well as each year of residency in Canada, in order to get the probability of infection. We then combine these probabilities with the 2016 Canadian census data to get TBI prevalence by age, by TB disease incidence in country of origin, and by year of arrival. So we found that overall estimated prevalence of TBI was approximately 20%, and 17% of this population had arrived within the last 5 years. Prevalence increased with age from 2% among persons 0 to 14 years of age to 32% among those who were greater than 74 years of age. Prevalence also increased based on TB disease incidence per 100,000 persons, with persons from countries within the less than 50 incidence band at 8%. And then this jumped to 34% among persons from countries within the 200 plus band. Among those 55 or older, you'll notice that the prevalence for those arriving from a country within the 200 plus band was observed to be lower than those in the 100 to 199 incidence band. This was reflective of earlier arrivals from countries whose incidence would have fallen into the 200 plus category prior to 2016. So this would be countries such as Vietnam or Ethiopia. In addition to overall prevalence, we wanted to look at trends. So we know that shifts in the demographic picture of Canada are associated with shifts in not only TB disease incidence, but likely TBI prevalence as well. So I wanted to see how the number of arrivals from high incidence countries had changed over time. Here we see that in recent years, a greater number of migrants have arrived from countries with higher incidence compared to prior years. In particular, we see that prior to 1975, arrivals within the 0 to 49 incidence band made up a greater portion. Then after 1975, that's when we start seeing a shift towards more arrivals from countries with 50 per 100,000 incidents. So we see a particularly sharp increase also in the number of arrivals who came from countries with incidence of 200 per 100,000 persons or higher. Then I went on to use this to also look at TBI prevalence by incidence band. When we look at this among arrivals in Canada for each year, we see that the prevalence was steadily increasing until around 1950. This was the peak for countries that fell into the 0 to 49 incidence band and the 50 to 99 incidence band. Prevalence then steadily declined among migrants from countries whose incidence fell below 100 per 100,000. Among migrants within the 100 to 199 band, we see a plateau in 1950 rather than the decrease that's seen in countries with arrivals from countries with lower incidence. And then we see a sharp decrease around year 1980, once again reflecting declines in TB disease in countries that previously would have fallen into the 200 plus category. So among those in the 200 plus incidence band, prevalence increased with subsequent arrivals until the plateau that we see around 1990, which continued into 2016. All in all, we see that prevalence was higher among earlier arrivals, especially those arriving around 1950. Prevalence was substantially lower among arrivals from countries within the 0 to 49 incidence band compared to other arrivals, suggesting that they may require less focus in TBI programs. And we also continue to see that prevalence is higher among migrants arriving from countries within the higher incidence bands, especially those within the 200 plus incidence band. Thus, that population may receive more benefit from TBI-oriented screening and treatment programs. Here we have the top 10 countries with the most arrivals. These are ranked from most to least arrivals. So we see that most arrivals in 2016 came from India and the least of the most came from Iran. We see that India, Pakistan, China, the Philippines, and Vietnam were countries with the greatest numbers of migrants arriving with Canada that also had some of the highest prevalence, that being greater than 25%. Among arrivals from Vietnam and Pakistan, prevalence was estimated to be around 45%, and that was the highest of these countries. So overall, this data suggests that we may have a substantial number of arrivals who may have prevalent TBI and who may benefit from improved programming. We also wanted to look at the relationship between time since arrival and TBI prevalence. As expected, overall prevalence increased with incidence, but we see that prevalence is higher among migrants who spent over 10 years in Canada, with the exception being those from a country with incidence greater than 200 per 100,000. Among this group, prevalence remains about the same regardless of how long migrants have spent in Canada. But overall, I think the trends that we see across these groups may reflect the fact that those it may reflect those with TBI who didn't progress to TB disease and have simply continued to live with it, or they may have cleared infection. Unfortunately, within our data set, we couldn't actually track who had cleared infection over time. So to conclude, we estimate that 1.4 million or 20% of foreign-born Canadian residents have or have had TBI. Our colleagues estimated global prevalence to be 23% back in 2014. And our results are also in line with prospective studies evaluating evidence of TBI using EGRA or TST. Thus, we expect that our estimates for foreign-born arrivals in Canada are likely fairly accurate. Note that these estimates do not account for possible clearing of TBI over time. And also we observed that prevalence of TBI increased with age, year of arrival, and with incidence in the country of origin. In particular, we observed that prevalence tended to be higher among earlier arrivals and arrivals from countries with higher TB disease incidence. Prevalence was also estimated to be higher among those of older age by 2016. We hope to be able to use these estimates to inform screening and treatment programs for foreign-born persons in Canada. So lastly, I want to thank my co-authors for all their help and contributions. I definitely couldn't have done all of this without them. And also I want to thank everyone for listening to this talk. And at this point, I will take questions.
Great, thank you. Thank you, Arya. Great timing as well. Right, questions from the judges or from the crowd? Marcel has a question, please.
Hi, thank you for that very clear presentation. Regarding the targeted screening and treatment, I was reflecting on Priya's discussion that her father goes back to, I think, South Asia several months per year. So have you considered the aspect that the foreign-born people are coming and going and how that would impact on any programs?
So with the data we have so far, we can't really, the short answer is we can't really consider that. We really, we had to take more of a blanket approach for this round. So we'll have to see if that can be considered in the future.
Thank you.
I have a question. So Arya, if you've read of some of Marcel's BMJ papers and just the natural history studies, we know that a few years after the infection, the risk of progressing to TB disease is very low, right? Much of the risk is concentrated in the first year, actually. So even though these numbers look like a large number of people have TBI, most of them will probably, it doesn't mean much, right? Because they will not progress. Is there any way of getting at the much smaller group that is likely to be at higher risk of progressing?
That's a good question. So we can see it. So one way to look at it is time since arrival. So we would be able to see for sure, who arrived within the last 2 years. So we could definitely look at that population. We can also go based off of what we know about certain individual risk factors. So we know that that if someone's under the age of 5, then they're going to progress a lot faster. Then mathematically, as of right now with this data set, not yet, but that's also something we could, I was actually interested in as well.
So just to follow up, Priya, do you have comorbidities, HIV, infection, diabetes, any of those in this data set?
Yeah, so those aren't in this model, but when we, basically the goal is, one of the goals for me is going to be to use this in a cost effectiveness analysis model. And for that model, we may be wanting to consider other subgroups. So we'd be applying it in that way.
Okay. Okay. And can I ask, out of the 217 countries, roughly what proportion did you rely on the Stiblo rule or modified Stiblo rule, and what proportion you could actually find tuberculin surveys to get annual risk of infection?
Oh, so unfortunately, I would have to get back to you on that.
Okay.
Yeah.
Erwin, please go ahead.
So maybe you said that. Thanks a lot for the talk, by the way, Aria. But I wasn't clear, what proportion of the TBI was determined with an IGRA type test and which proportion was determined with a TST type test?
Yeah, so this is all based on TST and Stiblo ratio, not on IGRA.
So it's all TST.
Yeah.
And Madhu, you had a question as well?
Is it possible to get actual numbers of people who are skin test or IGRA positive from Immigration Canada by country?
That's an interesting question. I will look into that.
Wouldn't that be a more direct estimate, given that all immigrants must undergo TB screening test?
Well, so all immigrants have to undergo a chest X-ray, but they don't necessarily have to undergo TBI testing. So it wouldn't. I don't think it would be accurate because we'd miss a a ton of people. So
agreed.
Right. Yes. No. Exactly. I think it's a very selected subgroup that get testing for TB infection. Very selected subgroup. So you'd have kind of really quite biased estimates of prevalence of infection.
Even that only post-landing or pre-landing?
That's all pre-landing, but post-landing is very heterogeneous. Depends where you go post-landing, what happens to you. And that too, post-landing is only 2 or 3% of all foreign-born arrivals. So again, it's a selected subgroup. Great. Okay, Aria, I'm going to close it there. Thank you very much. Great composure answering those questions. Excellent presentation. Thank you. Okay, so next, second presentation, pivoting now from the population level to the lab. So this is Kim Tran, who will be presenting on cross-protection of BCG vaccination in influenza infection, training adaptive immunity. Okay, so please go ahead, Kim.
Thank you for the introduction. So hi, everyone. My name is Kim. I am a PhD student in the lab of Dr. Maziar Divangahi. And as was mentioned, I'm going to be switching gears just a tiny bit because I don't actually work directly with TB, but I do work with the BCG vaccine, specifically investigating whether the BCG vaccine could cross-protect against influenza infection and whether we could train adaptive immunity. So as you all may know, the Bacillus calmette-guérin vaccine is a live attenuated Mycobacterium bovis vaccine currently used worldwide to protect against a disseminated form of tuberculosis in children. And even though it has been very effective in doing so, early on upon its introduction of the vaccine, there was a noticeable reduction in childhood mortality in the first year of life that wasn't attributed to tuberculosis-related death, which brought forth this idea that perhaps the BCG vaccine could be inducing a non-specific protection against a broader range of pathogens. And since then, multiple groups have gone on to show that BCG could protect against multiple different pathogens in murine models, including Candida albicans, schistosoma, and Plasmodium. And recently, the ability of BCG to cross-protect against a broad range of pathogens has been linked to its ability to generate trained immunity. So trained immunity defines that a primary stimulus, which could either be infection or vaccination, triggers the innate immune system and then returns the system to a slightly heightened basal state, which doesn't mean that the innate immune cells are constantly activated, but that the system is slightly more braced for infection, so that upon a secondary stimulus, we see an enhanced innate immune response. In trained immunity, it's this idea that innate immune cells have a memory-like capacity, which in comparison to the classical model of immunity, only adaptive cells typically have the ability to have immunological memory. But in trained immunity, through epigenetic modifications and metabolic reprogramming, innate immune cells are able to have long-term effects compared to their typical responsiveness of their short-lived innate immune functions. Central trained immunity involves the bone marrow, whereby vaccine or adjuvants that are administered systemically can access the bone marrow through the circulation, and there will affect the progenitor cells and generate trained innate immune cells, which will go back into the circulation, into the peripheral tissues, and protect against subsequent infection. Because the effect is seen at the progenitor level, the observed protection is generally much more long-lasting than what we would see in peripheral trained immunity. Our group has shown that when the BCG vaccine is administered intravenously, it can access the bone marrow and reprogram hematopoietic stem cells to enhance myelopoiesis and generate trained immunity. On the other hand, when MTB is in the bone marrow, it has an opposite effect and impairs myelopoiesis. It's also been shown in this study that was actually referred by Dr. Flynn earlier today, that BCG vaccine intravenously in non-human primates protects against MTB and is coincident with an expansion of T cells, which suggests that perhaps BCG could also be having an effect on lymphopoiesis. Because the progenitors of all immune cells reside in the bone marrow, the impact of BCG can't only be limited to the myeloid compartment. So we thought if BCG intravenously could access the bone marrow and generate an enhanced innate immune response, could it have a similar effect on lymphoid lineages and induce an enhanced T cell response? And of particular interest would be memory T cells, which could confer long-lasting protection. Such T cell responses are a critical component in antiviral immunity. So we sought out to see whether BCG IV could protect against influenza A virus, which is an important virus that is still responsible for thousands of deaths each year and still bears considerable pandemic potential. Protective responses against influenza A virus show that T cells target much more highly conserved regions of the virus compared to seasonal vaccines, which will generate antibodies against surface viral proteins that are very prone to genetic variation. So if we were to induce T cell immunity against influenza A virus, we could offer protection against a broader range of influenza strains and reduce the risk of outbreaks. So our hypothesis is that the access of BCG to the bone marrow will generate a heterologous T cell response that provides protection against subsequent influenza A infection. So first we asked, does BCG IV protect against an influenza? To do this, we vaccinated mice with BCG intravenously, BCG subcutaneously, and a PBS control. The BCG subcutaneous offers a route of administration that doesn't allow access to the bone marrow, but only to the lymphatic system. And we vaccinated the mice for 1 month, which is the time point at which our group has previously shown a peak expansion of hematopoietic stem cells. So we decided to stay with this time point for this study. So following 1 month of vaccination, the mice are subsequently infected with influenza. And at multiple time points, we look at several different readouts of infection. So first, we see here that BCG IV in red provides a significant survival advantage against influenza infection compared to subcutaneous and PBS controls, and also has an improved weight loss curve. When we look at the viral burden in the lungs of the mice, we see that at day 3, BCG IV actually significantly reduces by almost a log difference of the viral burden, and also slightly at day 1. We see that at day 6, the viral load starts to equalize, which suggests an early mechanism of protection. So to determine which branch of the immune system is responsible here, we first generated bone marrow-derived macrophages from vaccinated mice, and then subsequently infected them in vitro with influenza. and saw that there were no differences in viral load, meaning that trained monocyte macrophages aren't the responsible players here. We then went on to infect RAG1 knockout mice, which are lacking in T and B cells, and saw again the loss of protection that we had previously seen in wild-type mice, suggesting that BCG cross-protection is dependent on the adaptive immune system. And in fact, when we went back and looked at the adaptive immune responses in wild-type mice following BCG vaccination, we see that there's actually a drop in B cells, an increase in T cells. And when we checked for the influenza-specific antibody titers, we see no differences between the groups, suggesting that it's not the B cells, but likely the T cells that are the predominant player here. When we looked more specifically into the expansion of the T cells that was occurring in the blood, we saw that the expansion was actually due to an expansion of CX3-CR1 high T cells. So CX3-CR1 is typically cited as a monocyte marker, but recently there's been a new classification detailing the expression of CX3-CR1 for memory T cells, which defines 3 distinct populations of CX3-CR1 low, intermediate, and high. And we see clearly that in the BCG IV mice, there's a shift towards CX3-CR1 high, which is seen enriched in both CD8 and CD4 cells. To specifically assess whether these cells are also present in the lung tissue, we adopted an intravascular staining technique to discriminate between the lung vasculature and the lung parenchyma. To do this, we first intravenously injected an antibody of CD45.2 to stain all the vascular leukocytes, waited 2 minutes, collected the lungs, processed them, and stained them a second time with a different antibody of a different color that is also CD45.2, which will then stain the leukocytes of both the vasculature and the parenchyma. So that upon analysis, the double stain population will have undergone both the in vivo stain and the ex vivo stain and correspond to the vasculature. And the single stain population will have undergone only the ex vivo stain and correspond to the parenchyma. Using this technique, we found that in fact, the CX3-CR1 high T cells are also present in the lung tissue. We also found that the CX3-CR1 T cells are not antigen specific, as there were no differences in flu-specific T cells between the mice, and that the expansion of the CX3-CR1 cells weren't contributing to antigen specific responses, but that actually the CX3-CR1 high T cells have a heightened ability to produce interferon gamma and TNF alpha, suggesting that perhaps they are acting in a cytokine dependent manner. So we know now that BCG IV protects against influenza and that BCG IV generates these CX3-CR1 T cells, but are these CX3-CR1 high T cells responsible for the observed protection? To do this, we isolated by cell sorting the CX3-CR1 negative and positive T cells and adoptively transferred them into RAG1 knockout mice. And we saw that only the CX3-CR1 positive T cell adoptive transfer reduced the viral load compared to the no transfer controls. Following this, we used a CX3-CR1 GFP knock-in mice, which means that all cells that have CX3-CR1 marker has the marker replaced by GFP. So it's not a functional marker, but the cells are still there. And this knock-in results in a reduced recruitment of the CX3-CR1 high T cells into the lung tissue. We see here that in yellow, the CX3-CR1 GFP vaccinated mice have a much reduced level of the CX3-CR1 cells compared to the wild-type control. And the absence of these cells in the lung tissue led to a loss of protection against influenza. We see here the GFP mice compared to the wild-type mice that no longer have increased protection against influenza, which suggests that not only is the presence of these CX3-CR1 cells conferring BCG cross-protection, but that also the migration of these cells into the lung tissue is required for BCG cross-protection against influenza. Lastly, we show here that the BCG cross-protection against influenza is long-lasting, since we see that even Post 2 months vaccination, we show that there is still a reduction in viral load, as well as 3 months post BCG vaccination. And we also wanted to see whether this was specific to the influenza strain that we were using. So we used an H3N2 infection and saw again that BCG was protecting against this different influenza strain, suggesting that perhaps BCG has the potential to protect against emergent influenza strains, including highly pathogenic avian influenzas. So in conclusion, BCG intravenously generates the CX3CR1 high T cells in the circulation that are capable of trafficking into the lung tissue. And upon influenza infection, they will reduce viral replication independent of antigen specificity. And this has tremendous implication because of the incidence of TB. BCG is widely distributed, and it could also be used as a first line of defense against emergent pulmonary viruses. And obviously I can't say that sentence without addressing the pandemic at hand. And I actually started this work pre-pandemic, and upon seeing such a strong phenotype against influenza, we did look whether BCG could provide protection against SARS-CoV-2. And we recently published here with all these wonderful people that unfortunately BCG does not appear to provide protection against SARS-CoV-2, meaning that there needs more investigation into why exactly BCG is providing protection against some viruses and not others, and how we can specifically utilize it therapeutically. So I'd like to thank my supervisor, Dr. Maziar Divangahi, and all the members of my lab, and all of you for your attention today. Thank you.
Great, thank you. Thank you very much. That's an excellent presentation. Okay, so I'll open it for questions. I don't know.
I have a question.
Time is a bit short, but please, let's go ahead. Yeah, 5 minutes for questions.
Just one question. It was a great talk, really fascinating. Do you know, or can you speculate what cells the cytokines are working on to restrict the influenza virus?
Yeah, that's a good question. We've been looking into whether these T cells are acting by bystander activation, whether pro-inflammatory cytokines by innate immune cells from the influenza infection is activating these T cells. We think that perhaps there could be direct cytotoxic effect, or it's just an increased infiltration of inflammatory cells that are reducing the infection.
Thank you.
Okay, Erwin, want to go ahead?
I have a silly question. Why do these mice die?
The mice against influenza?
Yeah, why do they die? What exactly makes them dead, you know?
They gradually lose weight over time, which is why the weight loss curve is an accurate predictor of their death. We don't actually know specifically why. The fact that they lose weight, perhaps they're so sick they can't feed themselves anymore and they die of that. But that is what we speculate. They could be dying of respiratory failure and then not being able to feed themselves, losing weight, and then dying. We don't know which comes first, but yeah.
Thank you.
Okay, I'm just waiting for a question from Marcel, but if not, we're running a bit late, so I'm going to pass on. Thank you, Kim. Really, it was excellent talk and very good response to questions as well. Okay, so the third in this lineup is Jonathan Campbell, who's going to talk about low body mass index at treatment initiation and treatment outcomes in multi-drug resistant tuberculosis, an IPD meta-analysis. Take it away, Jonathan.
So you're going to have to forgive me. I'm in like rural Ontario on terrible Wi-Fi. So I'm going to try and do this. I'm probably going to turn off my video just so that I can present to you guys without any loss. Can you see everything?
Yeah, I can see it.
Okay, all right. I'll give you guys half hour of my vacation time to present to you today. Okay, video's off. So as Dick said, I'm Jonathan Campbell. I'm gonna also hide the video screen here. I am a postdoctoral fellow working with Dick and Kevin, and I'm going to, as Dick said, I'm going to present our individual participant data meta-analysis that was focused on evaluating the association of low BMI at treatment initiation and RIF-resistant TB treatment outcomes. So tuberculosis strains resistant to rifampicin, which is the most important tuberculosis drug we have available, are difficult to treat. Here on this graph are global success rates for treatment of RIF-resistant TB reported by the WHO. And you can see that persons initiating treatment for RIF-resistant TB have substantially worse treatment outcomes than persons initiating treatment for drug-susceptible TB. In 2018, only 59% of persons with RIF-resistant TB had treatment success, which is an absolute difference of nearly 30% when compared to those with drug-susceptible TB who have success rates approaching 90% globally. Mortality is also extremely high among persons with RIF-resistant TB, with 3 times higher risk of death among persons with this form of tuberculosis. This world map here shows the prevalence of low body mass index, defined as a BMI less than 18.5 kilograms per meter squared. And you can see that it varies widely by country, but globally 1 in 10 persons, or about 10%, have low BMI. And it's thought that 1 in 4 to 1 in 3 cases of tuberculosis are said to be attributable to low BMI. Low body mass index has been shown to be associated with TB infection and mortality in both persons with drug-susceptible TB and with non-tuberculous mycobacterial disease. However, its impact on mortality among persons treated for RIF-resistant TB and whether this effect is modified by different characteristics is much less certain. Therefore, we did a study using a large individual participant data set of adults with and without HIV infection who were treated for rifampicin or multi-drug resistant TB to evaluate the association between BMI at treatment initiation and end of treatment outcomes. The database we used in this analysis came from nearly 13,000 persons treated for RIF-resistant TB across 52 studies identified through systematic reviews and public calls for data through 2018. For the present analysis, we only included studies where the majority of participants had known BMI, and from these studies only included adults treated with individualized longer regimens with known body mass index, HIV status, and sex. After applying these inclusion criteria, 5,148 participants were included in our study. The excluded participants were more likely to be treated in upper middle income countries, be living with HIV, have more complex drug resistance patterns, and receive newer drugs, though this was largely driven by exclusion of data sets from South Africa because BMI information was not typically available. This is a table of characteristics of the included participants. And overall, 1,702, or exactly 1/3 of included participants had low BMI. You recall this is substantially higher than the global prevalence of low BMI than 10%. This table includes select characteristics among the included population that I wanted to highlight as importantly different. Overall, participants with low BMI were younger. They were more likely to be female, have HIV, be treated in low or lower middle income countries, have extensive disease based on smear and radiographic findings, and be previously treated for tuberculosis. The main goal of our analysis was to compare participants who had a BMI less than 18.5 kilograms per meter square at treatment initiation to those who had a BMI of 18.5 or greater for 3 outcomes, which included all unfavorable outcomes, which comprised death, failure, recurrence, and loss to follow-up versus success, death during treatment versus success, and failure or recurrence versus success. To correct for the large differences seen in the 2 populations that I showed you on the previous slide, we used exact and propensity score-based matching on demographic, clinical, and treatment-related factors to generate 2 highly similar groups. These factors included things like age, sex, country-level income, extent of disease, drug resistance patterns, and treatment received. We did logistic regression to estimate adjusted odds ratios for each outcome on this matched population. Now, this plot on the right that I'm showing you is not all that important to understand or to really read, but it's rather to show that prior to matching, and as shown on the previous slide, they are represented by the red dots on this graph. The two populations were very different from each other. You can see many red dots falling outside the dashed lines, which are boundaries set where we conclude that characteristics were similar between groups. So prior to matching, the mean difference in the proportion of participants with various characteristics, such as sex, extent of disease, previous treatment, as I'd highlighted before, were very different. However, after matching, represented by those blue dots close to that solid line in the middle, all dots were well within the dashed line, showing these characteristics were now highly similar between groups. And for many analyses, we retained over 70% of the included population. In addition to our primary analysis, we also had several secondary analyses where we evaluated effect modification of different factors on each outcome, the cumulative incidence of mortality during treatment, and the probability of mortality as a function of BMI. This graph shows the adjusted odds ratios for different outcomes across the total population, shown in the black circles, those without HIV, shown by the red square, and those living with HIV, shown by the green triangle. Note the log scale on the x-axis. Estimates to the right of the dashed line, which indicate an odds ratio of 1, indicate significant increases in the respective outcomes among those with low BMI. For example, for the total population dot at the top that you see, it says that unfavorable outcomes are much higher among people who have low BMI. So in our primary analysis, we found that the odds of unfavorable treatment outcome were 1.7 times higher among those with low body mass index as compared to those without. And this was consistent when we stratified by HIV, though non-significant among those living with HIV. You can see that mortality was the most significantly elevated, with those with low body mass index having 3.1 times higher odds of death, 3.1 times higher odds of death during treatment, and this remains significantly elevated even when stratified by HIV status. Finally, the odds of failure or recurrence were 1.6 times higher among those with low BMI. Like all unfavorable outcomes, this point estimate was consistent when stratified by HIV, though non-significant among those living with HIV. We assessed effect modification by HIV status, but did not notice any significant effect modification. What we did find was suggestive evidence of effect modification for other outcomes, specifically for mortality by country level income and sex. The increased odds of mortality among those with low BMI was 4.4 times higher for participants from low and lower middle income countries, while only 2.5 times higher for those from upper middle and high income countries. For sex, we found that females had 4 times higher odds of mortality when they had low BMI, while for males, they had 3 times higher odds of mortality. You can see these border on being less than less than 0.05, which we considered suggestive evidence of effect modification. Interestingly, we found no evidence of effect modification for the new and repurposed drugs, bedaquiline and linezolid. This graph shows the percent of participants who died stratified by BMI and receipt of bedaquiline and or linezolid. The gray box represents those with low BMI. We found that although the proportion of participants who died was halved when these newer drugs were used, falling from about 20% to 10% for those with low BMI, and from about 8% to 4% for others, the outcome disparities largely remained. We found no evidence of effect modification. Beyond these factors, we also evaluated several other factors for effect modification on the outcome of death and many other outcomes, which included previous TB treatment, drug resistance patterns, and extent of disease, and we did not observe any significant effect modification for these other characteristics. Switching gears, we wish to investigate mortality even further, and we estimated the cumulative incidence of death up to 24 months after treatment initiation for participants with and without low BMI stratified by HIV status. For these analyses, we used a competing risks framework and used our matched population, so that the two populations represented in these graphs are highly similar for all other characteristics except their BMI. The graph shows the cumulative incidence of mortality on the y-axis and the time since treatment initiation on the x-axis. For the participants without HIV, we see that the risk of mortality is constant throughout treatment. So it seems like participants are dying at a constant rate. And the cumulative incidence is 15% of people with low BMI without HIV have died by 24 months from treatment initiation, as compared to 5% of participants who did not have low BMI. When looking at participants living with HIV, the absolute difference in mortality at 24 months was approximately the same, 10%. Though note that very different Y scale where the proportion experiencing mortality is significantly elevated at about 30% of those living with HIV who were underweight and around 20% of those living with HIV who were not underweight. Next, we wish to investigate how the probability of mortality changed according to body mass index. We used generalized additive models and estimated the probability of mortality as a function of BMI stratified by HIV status. And this graph displays the probability of mortality on the y-axis and increasing BMI on the x-axis. As you can clearly see, we found that regardless of HIV status, probability of mortality was inversely related with BMI. That is, with lower probabilities of mortality as body mass index increased. So in conclusion, low BMI at treatment initiation is associated with 1.7 times higher odds of unfavorable treatment outcomes. The cumulative incidence of mortality was 10% higher among persons with low BMI, regardless of their HIV status. The probability of mortality was inversely proportional to low BMI, with lower rates of mortality among those with higher BMI. Though newer, more effective drugs can reduce overall mortality, taken alone, they appear unable to eliminate disparities in outcomes, with significantly higher mortality still remaining among people with low BMI. And taken together, it looks like interventions upstream of TB diagnosis that address the root causes of low BMI will be necessary to reduce overall mortality. And I'd like to thank my many co-authors on this paper and the collaborative group for the meta-analysis of individual patient data and MDR-TB treatment, who contributed data to this analysis, which made it possible. Of course, the FRQS, which provided funding that permitted me to do this analysis. I'll stop here, I'll stop sharing, and I'll turn my video back on. Thank you for your attention.
Great, thanks, Jonathan. Right on time. So we have a few minutes for questions before we conclude the this part of the afternoon. Questions? Okay.
Simon, you had a question?
Yeah, thanks, Jonathan. Can you hear me?
I can hear you.
Yeah, super. So my question is linked to your last take-home message. I'm just assuming that in all that literature, people mostly report BMI at treatment initiation. But from reviewing that whole literature, do you feel there's a signal there that reinforces our thinking that we should support nutritional status during therapy? Is there a measurable impact there that increasing BMI during treatment when we have patients within care structures increases success?
Yeah, that's a great question. And it's a question we couldn't exactly analyze given the data we had, which is only BMI at treatment initiation. But there is other literature, particularly for drug susceptible TB, that participants who have low BMI but gain weight during treatment have better outcomes than people who fail to gain weight, though randomized trials and observational studies of caloric repletion during treatment, so providing nutrition, have had mixed results. Some favor that to improve outcomes, some not. So that kind of led me to the conclusion here that while there's probably some evidence that people who put on weight during treatment do better, simply providing nutrition to people who are underweight at treatment initiation is unlikely to be the answer.
Okay, actually, I realized there were a couple of other questions earlier. So Marcel, actually, you were next, then Gonzalo, and then Erwin, and then I think that'll be it for the questions. Go ahead, Marcel.
I think my Wi-Fi is unstable, so I'm not sure if I'm asking the same thing. So let Gonzalo and Erwin go ahead.
Okay, Gonzalo. Gonzalo, you're muted, by the way.
Sorry about that. Thanks, Jonathan. That was a really good presentation. Great work, really. Two things. I mean, I think that the result of this work, first of all, hopefully will help folks that are doing randomized trials in this, that they should be stratifying on BMI when they enter these trials, because they could, make the outcomes look a lot worse than they are. And so I think that already, from the work that you presented, could have a really good impact. My sense is, in terms of thinking about what this might mean and whether you would consider this, is could it be that it's malabsorption of the drugs? So basically, you get someone with a really low BMI, and it's not, I mean, it's the BMI, but it's the fact that they can't absorb the drugs that then result in the bad outcomes.
Yeah, that's for sure a possibility, and one that we've seen kind of with first-line drugs, that persons who are underweight struggle to properly absorb them. I hadn't found any literature looking specifically at the newer drugs on this, but in the full paper that we've written on this, that does factor in as something that we speculate could be contributing to poor outcomes, is in fact drug malabsorption.
Thank you. That was really good. Thanks, John.
Great. Okay, Erwin.
Thanks, Jonathan. That was really great. So listening to Kim talking about metabolic reprogramming, right, being very important, and then listening to your talk, so I'm wondering, what do we actually know about pathogenesis in severely malnourished TB patients versus to normal guys, right? I know from my own experience, when we just take blood, we usually would not take blood from someone who is severely malnourished. So I'm wondering, do we actually exclude a very important patient population from follow-up studies? Do you know anything about that? Have there been on the molecular level, immunological or whatever, right? Are there any studies, that try to explain what you see?
In terms of unfavorable outcomes?
Yeah.
So, I mean,
just in general terms, the pathogenesis between malnourished people and regularly weight people. Because usually we would study regular weight people, which may be very different.
Correct. Yeah. So, I mean, low BMI is imperfect, but it is highly correlated with malnutrition.
Yeah.
And we know that people who are at which, and malnourishment impacts kind of your both innate and adaptive immunity, and results in protein energy undernutrition, which is crucial. Sorry, protein energy undernutrition is very important in terms of causing immune impairment, and it can affect other macronutrients as well. And we've had kind of co-authors on this speculate that there's many different factors that could contribute to it, but I don't think I've seen anything definitive on what particularly is causing poorer outcomes. One with the most data is protein energy undernutrition, and really that's influencing immune impairment. We have a co-author who thinks that leptin is very important, and in people who have low fat levels, that's significantly inhibited as well. But I have, really my in-depth review of this is that it's uncertain. There's many different theories on what could be causing it. I mean, we have like a whole paragraph on this written.
Talk to Mars about it and his metabolic reprogramming, right?
Exactly.
It's an overlap for sure. Anyway,
thank you for the question.
Okay.
Jonathan, did you consult with Anurag on this? Because I can't think of a bigger expert than him.
Right. So for this particular analysis, we did not, but his work was kind of what influenced me to take a look at this, and actually a lot of his previous work to conclude that really we're probably looking at the wrong place to correct malnourishment when somebody's diagnosed.
So maybe a
Because-
quick Zoom call with him will really help you nail this.
Of course.
I think he'll enjoy it, obviously, because this is exactly what he's been talking about. But we may be missing something given his big trial in India. Might be very, very valuable to have him on board.
Yeah, very interested in seeing the final results of that. I know you presented some interim promising results earlier.
Yeah.
Thanks.
Okay, so I'm gonna stop the question period. Okay, thank you, Jonathan. Please return to your vacation. If I could just get the three judges to make sure they send to Lysandra their grids. Madhu, you've sent, I guess, but judging by the thumbs up. So just send them quickly, and then we can announce the winners momentarily, assuming that there's no ties. It's kind of now my pleasure to announce the prizes. So I can go ahead with the prizes for best publications, because these were publications where a trainee was first author in 2021. So the Dr. Emil Scamany Prize for best publication by a trainee in biomedical research goes to Shannon Duffy, who published a paper in Microbiology Spectrum entitled Development of a Multiplex Real-Time PCR Assay for M. bovis BCG and Validation in a Clinical Lab. So Shannon, congratulations. I'm not sure if you're still here. We had 90 plus at one point, but now we're down to 73. So I know there's been some attrition. It's difficult on these Zoom calls, but anyway, okay. So Shannon. Okay.
Nick, do you want me to accept on her behalf and say, good work, Shannon?
Yeah, congratulations to Shannon, please. Okay. So next we have the Dr. Margaret Becklake Prize, actually in this case prizes, for best publication by a trainee for health outcomes research. So Ashna Upal, actually and Jody Boffa both won here. So Ashna's paper was on active screening for tuberculosis in high incidence Inuit communities in Canada, cost effectiveness analysis. This was published in the CMAJ, and Jody Boffa published quality of care for TB and HIV in the private health sector, a cross-sectional standardized patient study in South Africa. This was published in BMJ Global Health. So Ashna or Jody and/or, okay, their supervisors want to stand up?
Amrita is here. Jody's
Amrita's there, yep.
Congrats, Amrita.
I don't see Kevin, but congrats.
Yeah, then Ashna's in a meeting, but yeah, then Kevin's also on call, I think. So I don't think they're here.
Okay, so great. All right, we have all these hardworking people. That's the COVID impact, I think, still hanging on. Okay, so congrats to them. And then finally, the Dr. Jonathan Meekins Prize for Best Publication by a Trainee for Clinical Research goes to Mayara Bastos for the latent TB cascade of care among people living with HIV, systematic review and meta-analysis. This was published in PLOS Medicine, again in 2021. And there's Mayara. Great, we have a trainee who's can appear. Okay, congratulations.
Thank you.
Yes, okay, terrific. So congrats to all the trainees. Check is in the mail, so to speak, or will be in the mail. And yes, on top of the honors. So now the votes for best oral presentation. I'm going to turn on my chat and I'm hoping. How's it going with the counts, Lysandra?
While you're waiting, Dick, if I can just jump in, because I was one of the fortunates who did not have any conflict of interest with all those trainees. Yes. And I just want to point out that there was really a high quality of papers, and I really enjoyed reading everyone's.
Great. Okay. Yeah, I should actually thank the judges again, because yes, for publications, for abstracts, and everything, it's definitely, thank you, Simon, for sure. And thanks also to Olivia and Faz. I think Olivia was here, at least at one point I saw her name, and Faz, but I don't know if they're still here. Anyway, again, thank you to all the judges, as it is definitely the work behind the scenes. So the winner of, so in normal, it's kind of like, what is this? anyway, like all good prizes, we start with the third prize goes to, so Aria Ed Jordan wins the third prize. Congratulations, Aria. Really, again, your abstract was picked out of many, so certainly third check is in the mail too, Aria. So Jonathan came second. Congratulations, Jonathan, for your vacation efforts. And finally, Kim Tran wins the prize for best presentation. Excellent work, Kim. Thank you.
Thank you so much.
Okay, great. So, okay, so now I'll just kind of, that's it for the TB Research Day. In closing, I just again like to thank our speakers and trainees. Really outstanding presentations. The audience, obviously, for participation, questions, comments. I would like to acknowledge funding for the McGill TB Center. So things like the student prizes and so on come from these funds. From the McGill Faculty of Health Sciences, and also we get support from the Research Institute of the MUHC to the McGill TB Center and the Collaborating Center. I would also like to take a moment to thank Lysandra Lanes and Judith Belfi, who have worked tirelessly behind the scenes with numerous emails, sometimes begging and pleading for judges, etc., replacing speakers at the last minute. So thank you to those two, really, for a lot of hard work to get this together. And there's always some glitches, some lessons learned, so every year it gets smoother. And I really hope that next year we'll all be again in person. And really my last thought, again, that as we learned today, TB is the world's, now it's the #2 killer among infectious disease. For decades, TB was #1. And then the HIV epidemic relegated TB temporarily to #2. And again, tremendous medical advances in HIV. TB resumed its old position as #1 killer. So TB has been kind of bumped aside by COVID in many ways. I think we all hope that COVID will recede. Certainly mortality has decreased where in countries where vaccination has been available. So hopefully it'll be available in all countries. And but when that happens, really TB is going to go back to being the #1 killer again. And that's really not, it's not a very good record for those of us who've been working in TB for so many years. So clearly there's much to be done as a TB center. As members, we need to keep working, innovating, collaborating, talking, educating, listening, and relegate TB permanently to the bottom of the list rather than where it is now, near the top. Thank you, and see you all next year at the latest. Ciao. Bye. Thank you, Dennis.